Buen día, bom dia, buenos días a todos. Good morning. It's a great honor for me to be here opening this FALAN network 2021. First of all, I would like to say thank you to IBRO and to LARC because both associations support FALAN, okay? Many thanks to both. FALAN is now composed by Society for Neuroscience from Argentina, from Brazil, Chile, Colombia, Mexico, and Uruguay, full members, but also it has associated members as Peru, Costa Rica, and Cuba. Just wait to the end of this meeting and you are going to see new members. What is a great pleasure for us. But I would like to ask you if you know the word federation. What is federation? Federation is an alliance, a league, a union. And because of that, those are he is the FALAN mission, that is facilitate scientific interaction and exchange between its member societies and the individual neuroscientists. Also promote education and training in Latin America, across Latin America. Promote, understand, and involvement in neuroscience among the general public and decision makers. Promote discussion in animals research, women's rights in science, and science financial burden. That's the reason why it is called federation. It's for all of us. FALAN was founded in 2013, and the first executive committee of FALAN was, the president was Professor Rom von Bernard. They organized the first FALAN Congress in Mexico. The second and the third executive committee was, the president was Professor Osvaldo Uchitel. This committee organized the FALAN meeting, the second one in Buenos Aires, beautiful city. And we are, I am Elaine Del Bel, the president, but we have Patricia Casina from Uruguay, Gloria Cardona from Colombia, Belén Ergolien from Argentina, and Paola Hager from Chile. We are the fourth executive committee. We supposed to organize a FALAN meeting in 2020, but with the COVID pandemic, it's a difficult time. So to stimulate interaction between our societies and the members, we decided to organize this virtual meeting, this virtual activity. And so the FALAN meeting was postponed for September 2022. This is imposed by the COVID situation. But Latin America and Caribbean is one of the most dynamic and exciting locations on Earth. We have a diversity of nations, cultures, language, histories, geography, and ecosystem. We have a diversity of science systems driven by passionate, brilliant, creative, emerging, and established leaders. Wow, so the next days, we are going to share our passion for neuroscience. The FALAN committee he presented, we never meet in person, just virtually. We thank you for being here with us. Now we have the honor to present Professor Osvaldo Uchitel, the second president of FALAN. He's going to tell us about some FALAN history. Thank you very much and see you soon.
Okay, may I speak?
Yes, please.
Can you listen to me? I don't know if I am in the picture. I'm okay, great. So first I would like to thank very much Elaine and all the members of the executive committee of FALAN to give me this opportunity to participate in this inauguration of this FALAN 2021 conference. I'm very excited to see FALAN carry on these activities and at the same time working on towards the third FALAN Congress in Belém next year. FALAN is a young institution and therefore does not yet have the strength and economic independence that we will desirable. But we must all remember that building a society it is a very difficult task and requires effort and time. FALAN starts long before the first Latin American Congress in Bucios. From my point of view, it begins with the development of neuroscience societies in each country, in our countries. In particular, I rescue, because I'm Argentinian, the changes in the Society of Neurochemistry in Argentina and its transformation in society of neuroscience. And that allowed many young neuroscientists to participate. But those young neuroscientists in Argentina, Brazil, Chile, and every country in Latin America, they did not appear magically. They were born thanks to great professors. And I would like to go through a quick tour to honor those who had laid the foundations that allowed the flourishing of neuroscience in our region. So I would like to share the screen now. And I would like to know how to do that. I would like to share the screen. May I share the screen? Yes, share screen. Okay. 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 And then, okay, now. So I call this, I hope everything is, the name is The Birth of Neuroscience in Latin America, a quick tour. And as you know, those who laid the foundations to allow flourishing neuroscience in our region is the ones I want to honor. And if you talk about neuroscience, you have to start with Cajal. It's impossible to start with another man. So this is a picture of Cajal in taking the train to get his Nobel Prize with all his friends. And I'm talking about Cajal because although Cajal, he went to Cuba when he was young, but he didn't work as a scientist there. He never came to Latin America. He sent some of his students, some important students, they arrived to some of our countries. For example, one of his students was Clemente Estable, a neurohistologist. He worked with Cajal and he created later on in Uruguay the institute with his name, he carried his name. Also in Uruguay, there were other students of Clemente Estable, like Garcia Aust and Pepe Segundo, which were also very prominent neuroscientists in Uruguay. But also Cajal influence came to Argentina through Pío del Río Ortega. Pío del Río Ortega discovered the microglia, and he he built up a school of pathologists whose one of his members is Moises Pollack. So this is in the 40s. He died soon in Argentina, but before that, he also went to Mexico. And in Mexico, he had some other students that they worked with Pío del Río Ortega in Madrid, like Isaac Costero Tundanca, and also some Spaniards like Ramón Álvarez Bulla. He moved from Mexico from Spain to Mexico, and he was a professor in Simbestad for many years, very well known scientist, neuroscientist. And also Mexico was very prolific, and actually people like Arturo Rosenbluth and Cannon, they were physiologists, but I would like to strengthen Ricardo Miledi, who was my mentor in University College London, but also he went back when he was already old to Mexico, and he built up a new lab. He was still very young in spirit. Pablo Rudomín is also a person very well known in the spinal cord studies, and he worked in the Sinvesta in Mexico. So you see, there were many people in Mexico. But now let me talk about this guy, which is the squid. The squid was very important in the development of science in some countries. In the 50s, the squid was the model to learn how the nerve produce the action potential and carry the signals through the axon. So the squid was a matter of work in the Instituto Venezolano de Investigaciones Científicas, the IVIC, where I visited him in 1970, and they had beautiful, wonderful courses on introduction in neuroscience and in biophysics. And in that institute, Dipolo, which was a Venezuelan, and Boyer, which was an Argentinian, they worked together also with Carlos Caputo. They were people, they were really founding the basis of the function of the nerve using the squid. But also the squid was very important for the development of biophysics and neuroscience in Chile. And actually we are going to have Mario Luxoro in this institute, which is in Montemar. They did incredible work using this squid axon to learn the sodium conductance. And I want to mention all these Chilean guys, which are really great: Ramon Latorre, Pancho Bensalilla, Eduardo Rojas, and Cecilia Hidalgo, who is going to speak today. And actually, I'm very glad to mention the name of Cecilia, because there are not too many women in this history, but now we can see that there are plenty of women in the area of neuroscience. So I'm very glad about this takeover of the ladies of the work in neuroscience. And and actually in this Montemar place, I had myself took a course with Rojos, with Eduardo Rojas and Elaine, and I am here sitting, and all these were Chilean students that are now today, they are professors in Chilean universities. And so there were many other very important science like Joaquín Luco and Carlos Izaguirre and Fernando Rega in Chile. Classical papers of hypersensitivity to acetylcholine by Luco and Izaguirre in 1955. So you see, all these build up neuroscience in Latin America. And I have to go now to Brazil, a huge country and a huge production of scientists very well known. Some of them like Aristides Leao, who had a very, very important work on migraine and cortical sprain depression, and Elisaldo Carlini on the use of cannabis already at that time. And Rocha Miranda in a huge school of visual, of studying visual properties. And other people like, well, it's good to mention that Rita Levi Montalcini visited Brazil, and she worked for some time in Rio de Janeiro. And she was an outstanding, and of course, a Nobel Prize, as everybody knows. And I also mentioned Ivan Izquierdo, an Argentinian who went very early in his life to Brazil, is one of the most cited scientists in Latin America, I have to say. So there were other Argentinians in Brazil too, like Covian. So just to my country, I want to mention Eduardo Soto, Ranguel Caputo, Héctor Maldonado, who developed a very strong model of studying memory in vertebrates. And of course, this guy, which is a special person, which I have to stop one moment and say, I start with Cajal, and you know that Cajal, he proposed the neural hypothesis, and the neural hypothesis was based on his studies on the microscope. But under the microscope, under the normal microscope, the light microscope, it's not possible to see the separation between neurons, which was the base of the neural hypothesis. And that came with the study of De Robertis in Uruguay, in Argentina, and also in the United States, showing that there is a gap between the neurons and then really proving Cajal neural hypothesis. So all these professors, they really encourage us to later on to continue the education of neuroscience in Latin America. So there were many programs arriving, Latin American training programs, VLP programs, IBRO programs, many schools, which I just mentioned one here in the early time. I was in the middle of these two guys, Clemente Estable and Luco. This is in Uruguay in 1973. I am behind that gray bar. So they were really incredible schools which allow us to go ahead in the development of neuroscience. But one person, one very important person in all this story is John Nichols. And John Nichols, he really create many schools, he organized courses in Argentina, Uruguay, Brazil, Paraguay, Peru, Bolivia, Ecuador, Guatemala, and you name it, and also all around the world. So he is really on the basis of our development. So I want to show you just for fun. Here is Elaine Del Bel, and here is Romy, which unfortunately we don't have it today, probably because of a problem, a technical problem. But this is Romy, the first president of FALAN. This is myself, and here you can see Professor John Nichols. This is in Cusco in 2002, and here is another group of students in the Clemente Estable, and this is a course sponsored by the Latin American Regional Committee, which was very important for the development of the neuroscience in our area. And finally, the last person I want to mention is Carlo Belmonte, because Carlo Belmonte was the General Secretary of IBRO at the time we want to organize the second FALAN Congress. And he was really very helpful, and he fought a lot for us, and he got money for us to do the meeting. So I really want to acknowledge him. And so that's in our story, to have these meetings which are summarized in this picture. Really, there was a lot of work before that to create this neuroscience community, which is growing and growing. I'm very happy about that. So I wish a really successful FALAN 22, and I think that's enough for me. I think Elaine Del Bel. I want to thank Patricia Casina, Gloria Gardona, Belén Elgoyen, and the treasurer, which I miss now, the Paola, for their work. Thank you very much.
It is a great honor for us from FALAN to have you all in this meeting. We will have several lectures, important ones, very interesting. And also we have a special forum for discussion tomorrow concerning FALAN. I would like to invite all of you to come, where in this forum we are going to discuss the new conduct code and also some initiatives that are important to keep FALAN alive. Also, after we have the FALAN assembly, and it is important that all of you, the presidents, the societies of neuroscience that compose FALAN, to vote in the new committee that is going to start probably by the end of the year. So thank you very much for being here with us. And it's a pity that Professor Homi isn't here today. As Osvaldo told, maybe she had a technical problem, but I'm sure that in thought she's here with us. So congratulations to all of you that help us to organize this meeting. And let's wait for the next, the first plenary lecture. Thank you and see you soon.
Pablo, please. Adelante.
Hello, everyone. My name is Pablo López. I'm from Córdoba, Argentina. Today, we have the pleasure to introduce Guillermo Lanuza. He's the first speaker that will present the first plenary lecture from this 2021 FALAN virtual meeting. Guillermo currently holds a position as independent investigator from the National Council of Science and Technological Research, CONICET in Argentina. He obtained his PhD at the National University of Buenos Aires in 1999 in the field of experimental biology. Then he did a postdoc at the very important place, the Salk Institute, under the direction of Martin Golding. And in 2013, he started his own lab. He's now the group leader of the developmental neurobiology lab at the Fundación Instituto Cellular from CONICET. Today, he will speak about the spinal cord neurodevelopmental. He has, in the last years, very important publications in the field, so reports, development, change neuroscience, in particularly the last two papers from his laboratory identified a new neural population, the contacting serotonin fluid contacting neurons in the spinal cord, and he also determined the transcription factors that balance neuronal versus ependymal state in the spinal cord central canal. So, Guillermo, it's a pleasure to have you here. Go ahead. Before, I want to just comment that please, if you have any questions, write them down on the chat, and I will transmit to Guillermo at the time of the discussion. Thanks.
Hello, everyone. I hope you're well, and maybe sometime soon we'll have the chance to meet in person. So first of all, and before starting, I'd like to thank the FALAN and the Hello everyone, I hope you're well, and maybe sometime soon we'll starting. I'd like to thank the FALAN and the organizers of this meeting, this FALAN 2021, for the invitation and for the opportunity of presenting our work. And I hope that these 3-day meetings serve to foster interactions between these complicated times. So I think this is recording, so I will begin the presentation. And for myself as a developmental neurobiologist, I am interested in the basic principles of neural specification and how to make the right type of cells in the right place, the right moment, and the right numbers. So, and also how time and space are instrumental in the creation of cell diversity in the organization of the networks that control animal behaviors. So as you all know, the spinal cord is the most caudal part of the central nervous system in vertebrates and is essential to perceive and interact with our environment. So the circuits involved in perception and movement are spatially segregated within the spinal cords. Neurons in the dorsal horn receive inputs from sensory fibers, process that information, and relay it to centers in the brain, and also directly to circuits in the ventral spinal cord that are important in the control of movement. So these circuits in the ventral regions of the spinal cord comprise various types of excitatory and inhibitory interneurons that control the activity of muscle innervating motor neurons. So, and the extraordinary diversity of cell types that are found in the nervous system have fascinated neuroscientists from the times of Ramon and Cajal in understanding how different types of neurons are generated and assembled into functional circuits in the spinal cord. Is important to understand the physiology and the organization of the spinal cord. That is an important topic by itself. But also the studies in the caudal part of the neural tube have served as a pioneering model to understand the mechanisms involved in the specification of different types of cells and the acquisition of particular identities that have been applied to other parts of the nervous system. So to understand the source of cell diversity in the spinal cord, we have to go back to the early embryo at stages of the formation and the organization of the neural tube. And studies over the last 20 or 25 years have established that positional identity is a fundamental organizing principle governing neuronal subtype diversification. As an example, in the ventral neural tube, and this is a scheme of a transverse section of the neural tube at the level of the spinal cord, different classes of neurons, which are initially identified by the expression of different transcription factors, are produced at defined dorsoventral positions from progenitors that are in the ventricular zone. So, and this process of diversification of fates is initiated very early by inductive signals that are morphogens that are produced from adjacent centers. For instance, sonic hedgehog produced by the notochord and the floor plate, wind and BMPs from the roof plate and the dorsal ectoderm. And these signals pattern the progenitors that are located in the early neural tube. So these signals activate or repress the expression of a combination of transcription factors that are expressed following a dorsoventral patterning. This sets up a progenitor transcriptional code, and according to this model, the combinatorial expression of transcription factors define different domains along the axis of the spinal cord. And with each of these domains, in the ventral spinal cord from P0 to P3, generate motor neurons or different classes of interneurons during development. So according to these studies of gene expression in the developing neural tube, at least 13 classes of neurons have been described and are produced along the dorsoventral axis of the neural tube, and each of these populations of neurons are called cardinal populations. And more recently, single-cell RNA sequencing project have profiled also the cervical, thoracic, and lumbar regions of the mouse neural tube throughout development. And this data has essentially confirmed these basic principles of dorsoventral patterning in creating neuronal diversity. So as is shown schematically here, progenitors that are divided into domains express a particular combination of transcription factors, and also this is true for postmitotic neurons that also express several identity genes that are characteristics of each of these populations. So in addition, these RNA sequencing projects have also provided with complete gene expression data at single cell resolution. And this data is serving to better understand the trajectories of differentiation during development, and also has provided important insights in the division of each of these cardinal populations of neurons into different subgroups. So the definition of genetic and developmental difference have been very useful to classify neurons in the embryo, and also to establish their relation with neurons in the adult spinal cord. So the systematic mapping and characterization of each of these populations of interneurons and motor neurons have shown that neurons belonging to each of these cardinal groups of cells have some common properties in terms of their neurotransmitter, the axon projection, and the connectivity. So also, developmental genetics has been instrumental in dissecting the contribution of different classes of neurons to the circuits or cells that process sensory information and control movement. So for instance, V0 neurons and DI6 neurons are mainly inhibitory neurons that project to the contralateral spinal cord. And several experiments have shown that the activity of these neurons is important in coordinating the proper alternation of our left and right limbs during walking. So there are also other types of inhibitory neurons, GABAergic and glycinergic neurons, which do not cross the midline, but project ipsilateral and are important in other aspects of the functioning of these motor circuits by providing, for instance, recurrent inhibitions to motor neurons, setting up the rate or the speed of locomotion, and also provides the reciprocal inhibition between flexors and extensors that is needed for moving our limbs. So different to the cerebral cortex, in which time is important in creating neurons that populate the different laminas of the cortex, the importance of temporal patterning in the caudal neural tube was less clear until recently, with the identification of several genes that express following a temporal pattern. So these are temporal genes. And the expression of these genes subdivides the neurons according to their birth. So the information that we have so far is, this was analyzed for one population of interneurons and also for motor neurons, is that neurons that are produced during different developmental times have variations in these basic programs within each cardinal group of cells. And they also participate in different microcircuits that are important for the motor movement or the processing of the sensory information. But probably the most important contribution of time to cell diversity in the neural tube is the transition from a neurogenic phase to a gliogenic phase. In the mouse, around 13 days of development, there is a switch, and progenitors regions that early produce motor neurons and interneurons now begin to generate glial cells, astrocytes, ependymocytes, and oligodendrocyte precursor cells that are also produced following a dorsoventral patterning, which each of these late domains producing glial cells instead of neurons. So this neurogenic and gliogenic sequence is highly conserved throughout the animal kingdom, and these late phases of development have been considered to have lost the capacity to produce neurons. So in the following minutes, I will try to convince you that this is not entirely true. There is some neurogenesis during late phases of development that and that produce a particular type of neurons that and the specification of these neurons have a dedicated developmental program of development. So we began with simple experiments of birth dating experiments by applying VRDU at different stages of development. And this analysis indicate that there is an important contribution of Lane-board cells to the area of the central canal of the spinal cord. So as you know, the central canal is the remaining of the ventricle, is part of the ventricular system of the central nervous system at the level of the spinal cord, and is filled with CSF, with cerebrospinal fluid. So, and we were interested in this area because in addition to ependymocytes and tanycytes, the area also contains some neurons, a specific group of neurons that had been elusive in terms of their development and their role in mammals. These new central canal neurons are called cerebrospinal fluid contacting neurons. They are not new. They were discovered a century ago by Kolmer-Agdur, and they were called Kolmer-Agdur cells for some times in amphibians. They are present in all the vertebrates that had been studied so far, from the lamprey to primates. And also, although there are some differences in terms of the morphology of these cells in different species, they have a peculiar morphology with a thick dendritic process, apical process, that is terminated inside the central canal of the spinal cord and is in contact with the cerebrospinal fluid. So based on the morphology and the strategic location in the interface between the CSF and the parenchyma, these neurons had been proposed to serve as chemosensors and mechanosensors. So, and in support of this idea, more recent experiments in the lamprey and also in zebrafish have shown that these cells are sensitive to extracellular stimuli, changes in pH, osmolarity, and pressure. And also studies in the larvae of zebrafish shown that these cells are important in the modulation of locomotion, and also at least some of these cells are important in sensing bending during swimming in the larvae. So to get access to CSF senescence in the mouse and study their properties and their development, it was important to have specific markers for these neurons. Central canal neurons express the transcription factor GATA2 by using a GATA2 GFP allele. We were able to label these cells with GFP that show that how they are organized around the central canal, both in the lateral and in the ventral aspects of the central canal of the postnatal spinal cord. So, and in addition, these neurons express and selectively express the ion channel PKD2L1 that is a member of the TRP ion channel family, and as is shown here, is strongly accumulated in the terminal that is in contact with the CSF inside the central canal. So we confirm that these GATA2 PKD2L1 were truly neurons by recording GFP cells around the central canal. They show that they were able to fire action potential in response to depolarization, and they also have inward and outward voltage-dependent currents, and these experiments are in line with experiments that our colleague Raul Russo in Montevideo has performed. These central canal neurons express neuronal markers, beta-tubulin, and other markers of immaturity. And at least the majority, or 85% of the neurons around the central canal are GABAergic neurons. So regarding their development, a hint of their origin comes from the robust expression of the transcription factors NKX6.1 that is a characteristic of the ventral part of the spinal cord. Now, to define the origin of CSF-CNs in the mouse and assess to which of the known ventral embryonic population of neurons they belong to, we generated mice in which each of these cardinal populations of neurons was eliminated by using DBX1 mutants in GRAIL1 DTA mice, CHOX10 diphtheria toxin mice, FOXN4 mutants or NKX knockouts, which have in which each of these populations V0, V1, V2A, V2B, or V3 are missing or were ablated. We found a normal number of PKD2L1 expressing cells around the central canal. And these experiments indicate that CSF-CNs do not belong to any of the known cardinal classes of neurons, and as such should be considered as a novel embryonic population of spinal cord neurons. So next we analyze the time course of appearance of CSF-CNs during development by assessing the expression of PKD2L1 at different stages, and found that the first CSF-CNs are seen at E14.5, and with their number increasing over the following 2 or 3 days. So this is a late developmental time where astrocytes and oligodendrocyte precursor cells are already seen outside the ventricular zone. And consistent with this timing of neurogenesis of CSF-CNs, we found that the progenitors that give rise to these neurons are still proliferating late during development. These are VRDU labeling experiments that show that at E13 or E14, progenitors that produce CSF-CNs are still proliferating, indicating that, in summary, that central canal neurons are late-born neurons are probably the only type of cells that are generated, neurons that are generated during these late phases of development, and they arise from progenitors that are still proliferating in the transition from neurogenesis to gliogenesis. So we also map the progenitors that give rise to central canal neurons. So by performing a detailed analysis of the origins of CSF-CNs, we found that there are in fact 2 different sources of progenitor pool or progenitor pools that produce central canal neurons. And these are the late P2 domain and part of the domain that produces oligodendrocytes, and also the border between the P3 domain and the floor plate. So each of these progenitors produce 2 different types of CSF-CNs, and each of these CSF-CNs that are labeled with different markers are organized around the central canal in the lateral part or in the ventral part, and these neurons have been called CSF-CN prime or double prime. And the analysis at P0 show that some of the physiological properties of these neurons are slightly different. So as a first summary, there is a sequence in the production of cells during development of the neural tube. During the neurogenic phase of development, progenitors in the ventricular zone produce motoneurons and different classes of interneurons. After the neurogenic to gliogenic transition, these progenitors start producing glial cells, but also neurons of the central canal. So CSF-CNs are late-born neurons that are specified together with glial cells. So we also show that CSF-CNs are different to all the known classes of interneurons, and in support of this idea, recent single-cell RNA sequence analysis show that CSF-CNs are a separate cluster within all the types of cells that are found in the postnatal spinal cord. In addition, we found that there are two origins of CSF-CNs that map to the most ventral part or the intermediate ventral part of the spinal cord. And importantly, then these experiments imply that some late spinal cord progenitors have still the capacity to produce neurons. Now, in relation to this potential multi-competence of late progenitors, we focus on the transcription factor SOX9 that has been shown to play a fundamental role in astroglial precursor development. So in contrast to the sustained expression of SOX9 in astroglial lineage, its expression is quickly downregulated during the differentiation of CSF-CNs. So to assess whether there was a conversion in fates, we generated SOX9 conditional mutant mice. And in the spinal cord of these animals, we found an augmented population of PKD2L1 neurons. So, and interestingly, this increase in cell number was not restricted to the ependymal area, but also an important number of ectopic CSF-CNs were found in the parenchyma. So our interpretation of these results is that migrating astrocytes that fail to differentiate properly in the absence of SOX9 are differentiated into CSF-CNs, and these experiments also show that there is some plasticity in terms of the differentiation of progenitors or early or astrocyte precursors in the late phases of the spinal cord development. So among vertebrates, there are striking difference in the timing of generation of central canal neurons. In the mouse, rat, and chick, they are the result of late neurogenesis, as I showed you before. But in zebrafish, xenopus, and also in the lamprey, are the result of early neurogenesis, where CSF-CNs are produced together with primary motoneurons and even before other classes of ventral interneurons of the spinal cord. So this heterochrony in terms of the differentiations suggests a post-displacement in the genesis of CSF-CNs in amniotes relatively to the common ancestor, and may reflect different constraints or changes in the selective pressure that maybe is associated with the role of CSF-CNs in larva swimming. So we ask, how are late mouse progenitors endowed with the capacity to produce neuron, and how they escape from the gliogenic and ependymogenic differentiation programs that operate massively into these late progenitors of the spinal cord. And in looking for genes that could be responsible for the production of neurons during the late phases of development, we turn our attention to the BHLA transcription factor ASCL1, which is expressed in the spinal cord. And as you might know, ASCL1 is a proneural protein that is involved in neuronal commitment of progenitors in different regions of the developing brain, the adult brain, and also the peripheral nervous system. And by mapping ASCL1 expression at E13.5 with respect to different patterning genes, PAX3, NKX6.1, 2.2, or OLIC2, we found that the distribution of ASCL1 expressing cells at E13.5 matches with the positions where PKD2L1 cells begin to appear 1 day later. So there is a coincidence in time and space between the expression of ASCL1 and the neurogenic events that give rise to CSFCS. So to determine where ASCL1 expressing progenitors indeed produce CSFCNs, we took advantage of ASCL1 Cre-ER line that was used in combination with conditional tomato reporters, and that when induced with tamoxifen label the cells that are derived from these progenitors with tomato. And this approach showed that the vast majority of PKD2L1 neurons around the central canal around birth are labeled with tomato, indicating that central canal's neurons derive from ASCL1 late expressing progenitors. So next, we assess the role of ASCL1 in CSF programming or late neurogenesis. And for this, we introduced two different mutations to ASCL1. First, we generated ASCL1 knockouts, which lack the expression of PKD around the central canal. And we confirmed that it reflects the absence of CSFCNs by using different markers of this type of neurons. So in addition, we analyze ASCL1 hypomorphs that express reduced levels of ASCL1. And in these animals, a reduced number of ASCL1s matches with a reduction in the population of PKD2L1 positive cells. So ASCL1 in the mouse plays a fundamental role in instructing CSFCN identity, but a completely different scenario was found in zebrafish. So these are lateral views of in situ hybridizations in the fish that shows that CSFCNs are still produced upon downregulation of ASCL1 paralogs in the zebrafish. So these differences propose that concurrent with the delay in the developmental timing of CSFCNs along the evolution of vertebrates, ASCL1 might have been repurposed in the gene networks that drives CSF production in amniotes, when progenitors are massively committed to produce non-neuronal cells. So, however, ASCL1 is not only expressed around the time of the production of central canal neurons, but is also expressed earlier in the ventral spinal cord. So, and then to answer when ASCL1 is important for late neurogenesis, we design time window deletions of ASCL1. First, we took advantage of FOXN4 mutants. FOXN4 is a transcription factor that is expressed in some ventricular progenitors. And in these animals, lack of ASCL1 early, but the expression of ASCL1 is largely restored by E13.5. So interestingly, in these animals produce number numbers of central canal neurons, indicating that the early expression of ASCL1 is dispensable for the differentiation of central canal neurons. So, and conversely, we deleted ASCL1 in the gliogenic phase by using an inducible Cre-LOX-P strategy. And under these conditions, CSF-CNs were absent after deletion of ASCL1 at E13 in the mouse, phenocopying the ASCL1 null animals. And this, in summary, these experiments demonstrate that ASCL1 acting during late phases of spinal cord development is fundamental to initiate the specifications of CSF-CNs. To better understand the role of ASCL1 in CSF-CN development, we ask which is the identity that prospective CSF progenitors acquire now in the absence of ASCL1. So for this, we perform fate mappings by using the same strategy as before, but now in wild type or ASCL1 knockout background. So first, we analyzed the morphology of the red cells and found that in the mutants, red cells have a wider apical contact and rarely protrude into the central canal. And this is a morphology that is reminiscent of the morphology of ependymal cells of the central canal, or ependymocytes of the central canal. We analyze also the expression of several markers. Cells in the red cells in the mutants do not express neuronal markers or CSF-CN markers, but instead express several proteins that are typical or are expressed in cells, in ependymal cells that surround the central canal. So, and finally, we perform recordings of individual tomato cells by whole cell patch clamp, and in control and mutant animals. Red cells in control mice generate action potential in response to depolarizations and also respond with fast and slow voltage-dependent currents. So in opposition, so red cells in the mutant were able to spike and also have small currents, and were intensively coupled with their neighborings, that is a property of ependymocytes of the central canal. So in the absence of ASCL1, cells that are derived from this prospective progenitors display the morphology, expression markers, and property of ependymocytes. And and consistent with this neuronal to ependymal fate transformation, in the central canal, in the ASCL1 mutants have an increased number of ependymal cells, here labeled with FOXJ1, and results in an enlargement of the central canal compared to the control animals. So, as a summary, CSF-CN's progenitors become ependymocytes in the absence of ASCL1. ASCL1 confers the neurogenic capacity to late progenitors, and these late progenitors have the cryptic bipotency to produce neurons and non-neuronal cells. Second summary, as I showed you before, spinal cord CSF-CNs are late-born neurons that derive from ASCL1 expressing ventral progenitors. The delay expression of ASCL1 initiates CSF-CN differentiation and suppress ependymogenesis, and thus ASCL1 controls the balance between neurons and non-neuronal cells in the central canal. So, and as I introduced before, GATA2 is expressed in all central canal neurons, both belonging to the CSF-CN prime cluster, that is the dorsal cluster, or the CSF-CN double prime cluster, that is the ventral group. And in addition of expressing GATA2, these PKD2L1 cells also express the transcription factor GATA3. And now to define a more complete transcriptional program of CSF-CN specification, we analyzed the expression of this transcription factor and tested their functions. So first, and distinct to AACL1 that is expressed in progenitors, GATA3 and GATA2 are expressed upon the conversion in post-mitotic neurons. So GATA2 and GATA3 are downstream of AACL1, as shown here, are present in tomato cells that mark the cells that are derived from AACL1 cells. And if we analyze the time course of expression of these transcription factors for the dorsal cells and the ventral cells, we found that in the dorsal cells, the CSF-CN primes, GATA3 precedes the induction of GATA2, while in the ventral cluster, GATA2 and GATA3 have a delay expression and both are induced simultaneously. So based on these results, we propose trajectories of differentiation for the neurons belonging to the dorsal or to the ventral cluster with slight variation in the timing of induction of GATA3 and GATA2, where GATA3 is before GATA2 expression in the dorsal cells, while both transcription factors are simultaneously expressed in ventral cells. So in support of this general scheme for dorsal and the ventral cluster, as I show, GATA3 and GATA2 are expressed in AACL1 derived tomato cells, and also these transcription factors are genetically downstream of AACL1, as they are largely missing in AACL1 mutants. So we also produce GATA3 conditional mutants, and in these animals at E15.5, there is a failure in the induction of GATA2 during development, and also absence of PKD2L1 cells. So arguing that in the absence of GATA3, some of the cells fail to reach the final stages of differentiation. In contrast to this absence of GATA2 and PKD2L1 expression in the most dorsal part of the central canal, the differentiation of the ventral CSF-CNs appears to be normal. And this was confirmed around birth, where in GATA3 conditional mutants, there is a complete absence of CSF-CNs in the lateral part of the central canal, while the dorsal ventral cells are preserved. Now, since GATA2 is simultaneously expressed with GATA2 in ventral CSF-CNs during differentiation, and also GATA2 and GATA3 have been shown to have some redundant or compensatory functions in other cellular systems. We analyze also double knockouts. So, and these double mutants not only lack CSF-CN prime, that is a dorsal cluster, but also has a nearly complete absence of the ventral CSF-CN double prime. And these results demonstrate or suggest that in GATA3 conditional mutants, GATA2 expression is able to compensate for the absence of GATA3, but most importantly indicates that GATA2 and 3 global activity at post-mitotic stages of differentiation is essential for the acquisition of CSF-CN identity. So as a final summary, neuronal diversity in the spinal cord results from the composite action of spatial and temporal patterning. During initial phases of neurogenesis, different populations of neurons develop following a dorsoventral template, but also temporary dependent subprograms of differentiation. And I hope I was able to convince you that there is neurogenesis during late phases of neural tube development. And these neurogenic events produce CSF-CNs central canal neurons, and that are produced from two different pools of progenitors and develop along with glial and ependymal cells. And finally, that these neurons that are born late during development are organized around the central canal of the spinal cord. So it's still unclear which is the function of these neurons in the spinal cord of tetrapods, including mammals. So, and if in similarity to the larvae of the fish and the lamprey, they regulate motor behavior, or whether they have been co-opted to serve different functions. So, finally, we have identified a basic transcriptional program of the specification of central canal neurons that is composed by AACL1, GATA3, and GATA2. AACL1 confers neurogenic potential to late progenitors and suppress the differentiation of non-neuronal cells, with GATA3 and GATA2 acting downstream of AACL1, control the specialization of these neurons. So, and to finish, I have to thank the people who did the work. This was initiated by Janina Petraca in collaboration with Micaela Sartoretti in the identification of the origin of CSF-CNs in the mouse spinal cord. This was continued by Daniela Di Bella and Abel Carcaño that identify AACL1 as a key regulator of late neurogenesis in the spinal cord. And now is continued by Carla Campetella and Joselina Berti that are studying different aspects of the terminal differentiation of CSF-CNs and also the multipotentiality of late progenitors of the spinal cord. I need also to thank Antonia Marín-Burguín and Belén Pardi for their collaboration in several experiments, and also Matthias Hammerschmidt for the experiments in zebrafish. So this is all, and thank you very much for the attention.
Thank you, Guillermo, for this wonderful talk about the differentiation of CSF contacting neurons. Pretty wonderful data. So we are now open for questions. I had one actually, but you answered at the very end of the presentation. What was the role, which is still, as you said, unclear. So there are plenty of questions. We will start with the question from Gonzalo Olivares. He asked, he would like to know if the CSF contacting neurons progenitors respond to Sonic Hedgehog signal during development. Guillermo mentioned that 85% are GABAergic. What about the 15% remaining?
Okay, so so the first regarding to the first question and Sonic Hedgehog signal. So, so first, one of the main functions of Sonic Hedgehog during development in the neural tube is dorsoventral patterning. So we have done some manipulations in the cheek during embryonic development, and as it was expected, manipulation of Sonic Hedgehog signaling affect dorsoventral patterning, and as a consequence of that, there is an altered specification of this class of neurons. So, so one of the consequence of inhibiting, for instance, Sonic Hedgehog signaling is a dorsalization of the neural tube. So these neurons were still some were produced, but at different positions. So were at very ventral positions. So that was regarding the patterning. The other, we didn't test a direct action or sensitivity or these progenitors to Sonic Hedgehog. So, but it's very likely that they do, because it has been shown that even during late phases of development, sonic hedgehog is still acting on some of these ventral progenitors. So, for instance, it has been shown recently that the generations of OPCs requires an increase, a temporary restricted increase in sonic hedgehog sensitivity in these progenitors that are in the ventral spinal cord. So it's very likely that there is still a function beyond the establishment of the dorsoventral patterning that is essential to draw the template, the basic template for development. So that was regarding sonic hedgehog. So the second was, about the neurotransmission of these neurons. Yeah, I said that 85 were at least 85% were GABAergic, and this was based in analyzing the expression of GAD with the GAD1 allele that drives a GFP. But we did perform some analysis of both GAD67 and GAD65, and there is some GAD67 also. So it's very likely that both GAD1 and GAD2 account for the for all of these populations. We didn't see any other main or classical neurotransmitter being expressed in these neurons. So we think that probably they are all GABAergic. So there is one important topic that might be related to the function that they play. Is in the lamprey and zebrafish has been shown to express and produce several important peptides. With somatostatin and urotensin-related peptide. These are not expressed in the mouse. We have been looking for other potential active neuropeptides, but I don't have a definite answer. So there is regarding the neurotransmission also, there was it was described in zebrafish to transiently produce serotonin, and we intensively look for it in the mouse embryo, late embryo, and early postnatal embryo, and we didn't find any expression or any production of serotonin or any of the enzymes that are involved in serotonin production.
Okay, we have another question from Enrique Porcianchi. What happened to CETA contacting neurons after birth, and how they relate to the postnatal neurogenesis?
Okay, so this I show some analysis at postnatal stages. I think the pictures were only until P14 or something like that. So, but several other people have been looking at these cells. So they remain attached to the central canal in the adult. We and some other people have looked for a potential late production of postnatal production of these cells, considering that the ependymal area has been proposed to be an area where there is latent stem cells. But by doing regimes or BRDU application and in the postnatal animals, we could not find any hint of production during the postnatal life. So it looks to be only produced at least in normal or physiological conditions during late embryonic development, but the neurogenesis of these neurons is finished before birth.
Okay, Flavio Solesi, he made some comments and then a question. He said the timing on the generation of contacting neurons, particularly the heterochrony between amniote and anamniote, is very interesting. the fact that they seem to be relegated to the end in amniotes even after astrocytes suggests the role for notch signaling and a sort of default phase in this species. So then he questioned, what do you know on notch signaling in specifying these cells? And it could be the basis for a differential generation timing in anamniotes?
Yeah, okay, that is very interesting. The difference or the heterochrony that is seen in different vertebrates. So regarding notch signaling, we think or we speculate that the early production in, let's say, non-amniotes is very likely to result from notch signaling activity. It has been shown in the zebrafish that manipulation of the notch signaling is quite important in the generation of these neurons, and actually manipulations of the synaptic can manipulate the balance between primary motor neurons and CSF contacting neurons. So yeah, notch signaling is very important for the early production of neurons in this species. We think that notch signaling is still important late. So AACL1, for instance, is known to be an effector of notch signaling. So in fact, if in the developing embryo we abrogate notch signaling late during development, we find that AACL1, instead of having this salt and pepper expression there in the ventral progenitors, so the expression of AACL1 is more uniform. So we don't know the consequence of this, but yeah, notch signaling is very important for sure for the early production of neurons, and it is very likely to play a role also later for this restricted expression of the preneural genes in only some of these neurons and not in their neighbors.
Great. Then another question from Ernesto Miguel. Is there a similar population of CSF contacting neurons in the brain? Does their neurogenesis take place in later stages of development as well?
Again, can you repeat?
Yeah, if there is a similar population of contacting neurons in the brain, and does their neurogenesis take place in later stages of development as well? Okay,
so the second part, I think I answered already. So there is no neurogenesis in the spinal cord or in the epidemiology to produce CSF in the spinal cord postnatally. So there are in other areas of the brain ventricles have been described some cells having a similar morphology, so some similarities. But the production of these neurons is only found in the spinal cord and in the most caudal part of the hindbrain. So they begin to be produced at the end of the fourth ventricle down to the end of the central canal. It doesn't look to have a major production in other areas of the ventricles. Nevertheless, there is, and this is in the lamprey, and I don't think has been shown in other species. There is in the hypothalamus, in the lamprey, there is a population that is strikingly similar to these CSF contacting neurons. Actually, they receive the same name. And relatively recent work from Sten Gridler have shown that these hypothalamic CSF contacting neurons also express PKD2L1 and have some physiological properties in terms of the sensitivity that are quite strikingly similar to the properties that these neurons have in the spinal cord. But so far I don't think there's any clear demonstration that in the mammals, for instance, in the hypothalamus, there is cells that have these properties.
Okay, we have one pending question from Gonzalo Olivares. What is the outcome of ASL1 overexpression in other progenitors at late stages of neural development?
Okay, so well, that is a very important question, but for us was very difficult to answer. Considering that the specification depends on AACR1, one would predict that overexpression of AACR1 or ectopic expression in other progenitors would produce some of these neurons, and we could not perform those experiments because of the difficulty of expressing AACR1 in vivo and restricted to these late stages. But we intensively tried to do those experiments both in the late advanced stages in the chick and also in the mouse, and where we failed in performing. But the prediction would be that a sustained expression of AACR1 or the ectopic expression of AACR1 would drive differentiation of these late progenitors into neurons. And considering the other experiments, I would anticipate that we will produce an increased population of CSF-CNS. This is important considering that expression of AACL1 or other pro-neural genes like neurogenins or neurogenin 2 in particular has been intensively used recently for reprogramming. So these pro-neural genes have the capacity of transform, for instance, astrocytes or pericytes into neurons in different parts of the brain. The anticipation would be that it's very likely that the overexpression of AACL1 at late stages would produce neurons, and I would guess that the identity or the particular identity of the neurons would be the CSF-CNS.
Okay, we are running out of time. We have, I think, 1 minute left. But just a short question about you mentioned the pendulum fate of decontacting neurons in knockout mice for ASL1. Can they convert to other cell populations like OPCs, for instance, or oligodendrocyte progenitor cells?
Okay, so the analysis of the cells in the mutant that we track using the by fate mapping. So we didn't find any increase in ASL1 derived cells different than the ependymocytes in the ventral aspect of the spinal cord. But this cannot rule out that there are some alternative fates that are and the identity of some of these progenitors could be some astrocytes or some OPCs. We could not find any conversion into these fates. There is another topic related to this, is that some of these cells that we propose that are converted into ependymocytes. Some of the ventral population is converted in, I would say, ependymocyte-like cells because they are radial glia that is in the midline. But these were the fates that we found. So essentially, ependymocytes or radial glia of the midline, and we could not find any changes in the number of OPCs or astrocytes in the ventral part of the spinal cord.
Okay, well, Guillermo, thank you very much again for your wonderful talk. Thanks for the people for asking questions to make this symposium more dynamic. We now have a short break, and after that we will start with 2 symposiums in rooms A and B. So we see you soon.
Okay, thank you very much. Ciao, ciao.
Welcome to the FALAN Network in 2021. It's a great pleasure to me to be the chair of this symposium named the Cellular Mechanisms in Neuroscience. I want to especially thank Elena, Alejandra, Susana, and Mychael for being part of this activity by sharing with us their research. Remember that you may post your question at the live chat. We're going to answer one of them after each speaker's presentation. And at the end, we will have 25 minutes to answer other questions and for concluding remarks. Now, I'm happy to present our first speaker, Dr. Elena Avale. She's from Buenos Aires, Argentina. Elena is a biologist from the Facultad de Ciencias Exactas y Naturales, Universidad de Buenos Aires, where she also made her PhD. She's currently an assistant professor at UVA and independent researcher of CONICET and a group leader at the Instituto de Investigaciones de Ingeniería Genética y Biología Molecular in heavy CONICET. Her research is focused on the study of molecular and physiological basis of neurodegenerative diseases in tauopathies. Her lab is studying new therapeutic strategies in distinct models of neurodegeneration. The title of her talk is Post-transcriptional Regulation of the Microtubule-Associated Protein Tau, Functional Consequences and Therapeutic Perspectives. Welcome.
Hello, I'm Elena Avale from Buenos Aires, Argentina. I'm going to share with you some of the results we have been producing in my lab during the last years. Thank you for this invitation. So as you know, by the title of this talk, my lab is interested in studying the microtubule-associated protein tau. Particularly what I'm going to show you today is the results we have been producing related to the control of transcriptional regulation of the MAPT primary transcript and its implication in tauopathies. So to begin with, tauopathies are this group of neurodegenerative diseases which are related to the abnormal accumulation of tau protein in neurons. And in spite of Alzheimer's disease is the most famous tauopathy because it's the most common and prominent in the human population. AD is a particular tauopathy because in addition to tau accumulation, there is extracellular accumulation of A-beta. So we are going to focus today more in the role of tau and accumulation of tau intracellularly. So I want to mention that there are many other tauopathies which are known as non-AD tauopathies or primary tauopathies, on which the only abnormal protein that is accumulating is tau, right? So if we have a look at this accumulation in affected neurons by EM, this is the kind of neurofibrillary tangles that have been described many years ago, and we know they are composed of insoluble hyperphosphorylated tau. So depending on the brain nuclei primarily affected, tauopathies can be classified as more cognitive behavioral, like dementias, like Pick's disease or frontotemporal dementias, or those tauopathies that manifest primarily motor impairments like PSP, progressive supranuclear palsy, or CBD, corticobasal degeneration. Yes, and these are sometimes known as typical Parkinsonism. I will show you this later in the talk. So to understand what happens in an affected neuron on which tau is not properly working, in this cartoon, this is very illustrative, I find it very didactic, is that in a normal neuron, we need a lot of cargos and molecules to move from the soma to the terminal, and this requests a very healthy and well-stabilized axon with microtubules where all the cargos will move. And we know from many years ago, it's known that tau has a very important role in stabilizing microtubules. But this is not the only role of tau. Tau has many functions. So I don't have time today to explain all that, but let's keep in the tau dogma for the moment. But we have to know that tau is also very actively involved in the movement of particles through the axon. But well, when tau is detached from the axons, we don't know exactly all the reasons why this happens. This might be due to mutations, due to isoforms imbalances, due to hyperphosphorylation, or many other post-transcriptional or post-translational modifications that can go wrong. And thus, this will lead to the detachment of tau, the formation of tau deposits, and of course, microtubules will collapse, and this will be the consequence for the neuron. Of course, neurons will die. So we are going to focus today in this part of the tau protein. In this cartoon, you can see that there are many other parts of the tau protein that are involved in different functions, but particularly for the binding to microtubules, we are going to focus on these 4 microtubule binding domains. And now I want you to pay attention to the color code, because here we have the MAPT gene, yes, which is the gene encoding for tau. And these are exons, the 2 and 3 exons are coding for the N-terminal domains, and these are alternative spliced, and also the exon 10, which is coding for the second microtubule binding domain, will follow alternative splicing. So this means that the combination of alternative splicing of exons 2, 3, and 10 will give rise to 6 different tau isoforms in the human brain. This is in the normal human brain. But if we concentrate in this part, as I told you, which is the part involving binding to microtubules, it's very interesting to understand how the second microtubule domain encoded by exon 10 is alternative spliced and highly regulated. And this gives rise to 2 main groups of taus, which we call 3R or 4R. You can imagine now that 3 is because 3 microtubule binding domains, and 4 is 4 microtubule binding domains. So from now on, when we say 4R or 3R, we are referring to this kind of proteins containing or not the second microtubule binding domain. So the splicing of exon 10 is very, very well regulated in neurons. I'm not going to enter in detail with this. I'm fascinated about this, but we don't have time. So just the take-home message is that there are many regions, either in exon 10 or in intron 10, following the exon 10. Here, this is the intron, on which different splicing regulators will bind and will either increase or decrease the inclusion of exon 10. So given a constant sequence of a transcript, because this will not change through life, a neuron can decide which splicing regulators will produce in any moment of development, and that will give rise to a change in the type of isoform produced in the brain. And this is exactly what happens during development, either in humans or in mice. In early stages of development and early postnatal stages, only 3R proteins, 3R tau proteins are produced, because exon 10 is always excluded. But during development, in the case of mice, everything goes to 4R. It means that exon 10 will be always included. But in humans, we are always a bit more complicated. We have this equal balance between 4R and 3R proteins production. So basically, that's what happens in a normal mouse or in a normal human. But in humans, we also have this tricky thing, that is a lot of mutations that have been reported either in exon 10 or in intron 10 that will lead to a deficit in the control of alternative splicing. So what could happen, all these are mutations that have been related to tauopathies. These are human pedigrees that have been analyzed, and it was reported that concomitantly to the phenotypic manifestation of dementia or motor symptoms or whatever the clinical manifestation, they produce an imbalance of tau isoforms. So we are interested, I started this when I was a postdoc, given this problem that exon 10 splicing leads to disease, and there are more than 30 mutations in the MAPT gene that were related to tauopathies due to problems in alternative splicing. The idea was to modulate exon 10 inclusion or exclusion and try to correct this isoform imbalance. And to do this, during my postdoc, I've been spending lots of years trying to develop an RNA transplicing strategy to target the endogenous MAPT transcript. So I will summarize just for you to follow the talk. This is the endogenous, endogenous MAPT transcript. And as I told you, if everything goes fine in a normal brain, we will have an equivalent inclusion or exclusion of exon 10, and this will produce 3R and 4R proteins. Everything in gray is endogenous pre-RNA, splice RNA, or protein. By this system of transplicing that is quite sophisticated because we have to produce these pre-transplicing molecules with specific features. This is RNA that has to be endogenously produced in a given neuron. We do this by lentiviral vectors. But the idea behind this is that we can cheat the endogenous system by producing these exogenous molecules of RNA. So when splicing, when the endogenous splicing machinery will recognize the donor and acceptor sites, it will create this chimeric RNA between the endogenous MAPT RNA and our molecule. And in this molecule, if we include exon 10, we will force the system to produce this 4R RNA and the 4R protein, right? And conversely, if we decide not to include exon 10 in our construct, we will have the 3R protein, right? And this is chimeric, so everything in yellow is produced from our construct, the one that we introduce in neurons, and in gray is coming from the endogenous tau transcript. So I will skip this because it's just the control. If someone is interested, we can come later to this because we have, in those times, I did a lot of work trying to demonstrate that the splice junction was fine and the chimeric protein was produced and everything was working apparently fine. So then when I came to Buenos Aires and started my lab with my student Sonia, we decided to go and investigate, well, okay, this system is working nicely, but we want to try to make it work to correct a problem in a brain, in a whole mouse, and try to see if we can revert or prevent phenotypes. So we took advantage of this mouse, which was produced many years ago. This is an H-tau mouse. It's a knockout for the endogenous murine tau, but it contains a huge transgene in a PAC construct, containing the full construct with the human tau gene, all exons and introns and regulatory regions. So it's very interesting because this mouse has no mutations, but it produces tau pathology in the cortex and hippocampus and cognitive deficits from 9 months. And what was proposed at those times, and then we verified this ourselves, is that the problem of these mice is that the human transgene in the mouse neuron, combining the human sequence, but with the mouse splicing factors, this leads to an imbalance towards 3R protein production. And I told you at the beginning that in a mouse, the normal condition is to have 4R tau. So this might be the cause of these phenotypes, similar to what happens in humans, right? This is a model. We don't have exactly what happens in a mutation in humans, but it recapitulates quite well the phenotypes of these tauopathies, human tauopathies. So our first objective with Sonia was to try to find out which areas were more affected and try to test our system there. So we found in the prefrontal cortex the most prominent accumulation of hyperphosphorylated tau, which was also consistent with the phenotypes we were observing in these mice. So our question was very specific, and we said, okay, can we preclude tau pathology if we change this isoforms imbalance specifically in the cortex? And this is what Sonia did during her PhD. She injected quite a few mice with these constructs containing the tau PTM, which could exert a change in tau splicing with its controls as well. And after 6 months, 6 months after the injection in the prefrontal cortex, we found that the mice that have been injected with our transplicing molecules had a less prominent accumulation of hyperphosphorylated tau. This is quantified here, either with a CP13 antibody detecting early phosphorylation or late phosphorylation, and also by Western blot. We have seen that even if we do not go to the wild type here, but yes, here, the amount of hyperphosphorylated tau was reduced. In collaboration with Juan Belforte, we were able to characterize the firing pattern of cortical neurons in H-tau mice. This was reduced compared to wild types and recovered in injected mice. And also, we did a lot of behavioral analysis, and this is the most eloquent, in my opinion, is the recovery of normal phenotypes in the novel object recognition test. And well, this is what we reported in 2018 with Sonia and all the rest of the group, claiming that if we were able to correct tau isoforms imbalance early before the onset of disease, we were able to preclude or prevent the development of abnormal phenotypes. But then the story goes on, because we were also interested in analyzing what happens with with phenotypes related to motor function, because during examination of the H-tau mice, the girls in the lab, there were 2 girls at those times, they detected they were not that good when swimming or when walking. So since 2000, it was known that deficits in exon 10 splicing were related to PSP. And in 2019, it was published already a meta-analysis study on which it was well characterized that a tau isoforms imbalance leads to striatal tau accumulation and deficit. So my student Anna, who is now almost a doctor as well, started to do this motor coordination analysis in our mice. And she compared 3 groups in this case. These are wild types, knockouts, because mice lacking tau had been reported to have some movement disorders, and the H-tau. And this is the number of falls, the latency to first fall, and the maximum time in the road. And as you can see, the H-tau are really very bad. And these could be related to different problems, but basically what we started to think is that, okay, there is a problem in the neurostriatal pathway. So this could be either at the level of the substantia nigra, where the cell bodies of dopaminergic neurons project to the striatum, right? So Anita did a lot of experiments, either by immunohistochemistry or HPLC, but to our surprise, H-tau mice had no reduction of dopaminergic neurons, neither a reduction in dopamine levels in the striatum. And then Javier, another member of the lab, did a pharmacological analysis trying to see if the H-tau mice were responsive to L-DOPA, and they were not. So we concluded that the H-tau mice had normal dopamine contents, and the motor coordination was not rescued by L-DOPA, so this could be a typical Parkinsonism. So to try to find out where the problem was, we had the collaboration of a great group in Plenary here in Buenos Aires, in Escobar, Germán Falasco and his group, and with micropet, detecting glucose metabolism, we were able to find out that the dorsal striatum was showing hypermetabolism in H-tau mice. And with Camilo Minini from IBIME, we decided to go for an electrophysiological analysis, and it was real that the medium spiny neurons were firing less frequently than the wild-type neurons in the H-tau. And it was like a puzzle, because this was hyperactive or hypermetabolic, but there was less firing of these neurons. And in addition, we could not find any tau deposits in the striatum. But we were able to detect that in the striatum there was a huge imbalance of tau isoforms. So this is what we had at some point, and we're a bit amazed, because how can we have this huge deficit in addition to the deficit in motor coordination, but no tau deposits at all? And then, well, Mariano Zoisa-Reilly allowed us to put another piece of this puzzle, because he showed us that in these mice, in the H-tau, there is more corticostriatal connections, which could explain the hypermetabolism we see here. Maybe this increase in glucose uptake is not by the medium spiny neurons themselves, but by the cortical projections coming from the cortex, trying to compensate the lack of firing of the MNS. So this is what we concluded at those times. MSMs were not firing okay, so increasing cortical inputs and increasing fluorodeoxy glucose uptake. But well, the main thing here was to try to rescue these Parkinsonian phenotypes. If everything was pointing towards a problem in the striatum, okay, let's go and recover normal tau isoforms in the striatum and see what happens. And this is what we did. Anna injected all the mice, and then Carolina Facal did all the molecular analysis, and we were able to rescue tau balance in the striatum as we had done before in the prefrontal cortex. And surprisingly, mice recovered normal activity in the rotor rod, and they recovered or partially recovered a normal firing of medium spiny neurons. But bear in mind that I told you that all these phenotypes that we have seen and which were rescued by isoforms normalization were in the absence of tau deposits. So the take-home message is that this dogma of tau pathology always linked to hyperphosphorylated tau deposits is maybe not such a dogma. Okay, so this is the conclusion. So we had these nice pictures from Javier from the lab. So isoforms imbalance in the striatum leads to phenotypic deficits. When we correct firing, and mice are happy and very well coordinated. So this is my group. I have to especially thank well, the people of my group, of course, Sonia, who is now in Misiones, Anita, Caro, and Javier, who were part of this work with a lot of energy, and our collaborators in this project, Juan, Camilo, Juan Belforte, Centro de Imágenes Moleculares de FLENI, which are great people, Mariano Soizarelli from IFIBINE, who helped us to do all the characterization of the striatum, and the people funding us, hopefully for more time, because we want to go on with our project. Okay, well, thank you very much, and I'm open to all your questions. Bye.
Thank you very much, Elena. That's a very nice talk, very interesting work. Let's see if we have any question to answer. Yes, we do have a question. So let me translate this. What are the consequences of the cellular level of balancing the 4 error levels? Intracellular transport at the neuron, maybe? What do you think?
So, hearing me, I have a problem because I'm hearing myself in delay, and okay, well, that's a good question. Maybe someone from an associated group has been asking that. We have been testing that. I had no time to show all we have done, but one of the first things we have done before moving to the in vivo experiments, it was to actually change isoforms balance 3R, 4R in human neurons in culture. This was done in collaboration with Tomás Falzone. And we tested that imbalance, induced imbalance by our transplicing system, was actually affecting axonal transport, either in rank lengths or in the velocities of molecules moving through the axon. So yes, there is a huge effect of isoforms imbalance on axonal transport. It's published in 2017, so you can go and have a look, and really, well, it was something that was like, for us, was not such surprising, because we know that tau binding to microtubules would affect transport, but it was quite evident, more than what we expected.
Okay.
I don't know if that answers the question.
That's okay. So there's a question from Maria Luis Mourinho Ramirez. She's asking for some advice for the practice to give for patients treatment. That's a
that's an amazing
step.
question. Well, there is a big issue with patients treatment. I have to say that there are other systems which regulate splicing, particularly MAPT splicing at exon 10, that was developed a few years before our system in the US by Tim Miller's group, and it's based on ASOs, right? Antisense oligonucleotides that will go and interact with the splicing regions. And as far as I know, they are associated with a company, and they are launching, or they already launched clinical trials to try to correct tau or isoforms imbalance in patients. The problem, either with ASOS or with our system of transplicing, or whatever system you use to correct something in the human brain, in my opinion, is that so far, all our evidence is that this should be done very early. So we need an early intervention to avoid the accumulation of abnormal tau species. So practically, this means that a patient has to be diagnosed very early, maybe at the prodromal stages of disease, before any clinical symptom is there. So let's imagine a PET scan on which a doctor can detect in a patient that, okay, you are having accumulation of abnormal tau in this area, so probably you will develop dementia. And in that case, the patient has to be injected with these molecules that will modify splicing. So this is something out of our scope, but talking and having chats with neurologists led us to think that the main problem is there, because this is preventive and not curative. But well, so far. So now we have some data in the lab. One of the projects is to try to find out which is the therapeutic window for intervention. So in this same model in mice, but we are trying to, well, okay, let the mice start develop some of the phenotypes that will indicate that they are going to develop tauopathy, and then we interfere with our transparency system. And as far as the results we have, I'm not sharing that today because it's ongoing, but the outcome is not as good as the one we had with early intervention, because all I have shown today is injections we have done at 3 months. It means before the animals develop phenotype, so it's preventive. Okay, I don't know if that answers the question.
That's okay. That sounds challenging also.
Yeah,
so
it is.
you have more questions? Let them for at the end discussion. Now we are moving to the next presentation. So our second speaker is Dr. Alejandra Alvarez from Santiago, Chile. Alejandra is a biochemist from the Pontificia University Católica de Chile, where she also made a PhD. Currently, she's an associate professor and a group leader at the Departamento de Biología Celular y Molecular, Facultad de Ciencias Biológicas at the same university. Her group investigates the molecular mechanisms and signal transduction pathways involved in the degeneration of neurons, and focus on determining the participation of ABL kinases in the neuronal function and the potential role of CABL kinase as a therapeutic target for Alzheimer's disease treatment. The title of her talk is ABL kinases at the crossroads of healthy synaptic remodeling and synaptic dysfunction in neurodegenerative diseases. Please, Alejandra, welcome.
Okay. Good morning. First of all, I would like to thank to the FALAN Networking 2021 meeting organizer by inviting me to share with you our result about the CBL kinase and its role in healthy synaptic remodeling and its role in synaptic dysfunction. CBL is non-receptor tyrosine kinase that belong to the family of SAN-ARG family and have tyrosine kinase domain, SH2 and SH3 domain to interact with its target, but also have a big carboxyl terminal domain where this kinase have a domain to interact with actin, DNA binding domains, and have signal to import to export to the nucleus. Thus, these kinases have a wide range of functions, including gene expression, cytoskeleton dynamic, apoptosis, depending on its localization. CBL is a ubiquitous protein, but is highly expressed in brain. It's expressed in neuron, in the soma and nucleus. It's expressed in dendrite and axon, especially concentrated in row cones, and in mature neurons is present in presynaptic and postsynaptic compartment. Thus, the reported function of the CBL in neurons include neurogenesis, neuronal migration, cytoskeleton dynamic, axonal pathfinding, dendrogenesis, synaptic plasticity, gene expression. The most of CBL reported function are related with, or were related with, cytoskeleton dynamic and development. But some years ago, we identified CBL signaling as a key event that mediates Alzheimer's disease neuronal pathology. In neurons exposed to a beta peptide, as well as in Alzheimer's disease mouse models, we found CABL signal is activated. And so we found that this signaling of the CABL is involved in cell death, A beta production, tau phosphorylation, and how I will show you now, is involved in synaptic loss and dysfunction. In Alzheimer's disease, the synaptic dysfunction, synaptic loss, underlie to the cognitive decline. And CABL is a synaptic protein and is activated. The synaptic CABL is activated by A beta oligomers, as I show you here, where you can follow the CABL activation as its phosphorylation, and you can observe the increase of active CABL in the dendritic spine. Also, if you isolate synaptoneurosomes and expose this to A beta oligomers, you can observe the CABL activation. Then CABL signal is activated in synapses by A beta peptide, and more important, this activation mediates the synaptotoxicity. Indeed, when you expose wild-type neuron to A beta oligomers, you can observe a decrease of dendritic spine. But if you treat with the beta oligomers, neurons are null for CBL, this decrease in synaptic and the dendritic spine is reduced. More important, when we evaluate the synaptic contact, here followed as Piccolo 95 cluster, you can observe that the neurons that are null for CBL are protected against A beta oligomers. Then the CBL deficiency provides synaptic resiliency against A beta oligomers. Next, we generate Alzheimer's disease mouse model that is null for CBL by crossing ID mice with mice flock-set for CBL and stick-ren animals. We obtain the different genotypes, and these animals were evaluated, cognitively evaluated here in water maze assay following memory flexibility protocol. You can observe that the Alzheimer's disease mice model or mice, T mice, show takes several intents to find the platform in different days. More intent than wild-type mice, but if you use, you evaluate the ID mice, but null for CBL, these mice show a similar performance that, to wild-type animals and need less intent to find the platform. A similar trend we observed in Barnes maze test, where the animals that are null are ID mice, but null for CBL learn, while the ID mice have a poor performance. Then the lack of CBL improved the cognitive performance in ID mice. These results show us that CBL is involved in A-beta induced pathology. This function, CBL function, has been now reported, is activated by several or different type of damage. CBL in neurons could be activated by oxidative stress, beta-amyloid, lysosomal deregulation, ER stress, and this CBL activation is linked to neuronal death, synapses loss, cytoskeleton alteration, cognitive and motor decline depending on the disease. Thus, the emerging view of CBL is this is a tyrosine kinase that is a damage response integrator that participate in Alzheimer's disease. We show it is involved in Niemann-Pick disease and Gaucher disease, ALS, Parkinson, and now I will show you some recent results in epilepsy. Epilepsy is a disorder related also with neuronal synaptic dysfunction and neuronal damage. In the literature, there is only one paper that link the CBL activity with epilepsy. This paper of the 2014 that showed that in epilepsy patient brain, in the hippocampus, CBL is activated. Here followed as phosphorylated CABO. And also CBL was activated in temporal lobe epilepsy model in rats, where CBL was increased after pilocarpine injection. Then we evaluate the role of CBL in epilepsy, and first we used in vitro epilepsy excitotoxicity in vitro models. Here I show you that neurons exposed to excitotoxicity by MDA or glutamate show activation of CBL, and this activated CBL goes to the nucleus. There are an increase of the CBL in the nucleus, where this tyrosine kinase regulate gene expression involved in apoptosis, and this activation could be prevented by imatinib or GEMS2 inhibitors, CBL inhibitors. More important, we observed that the CBL inhibitors, imatinib or GEMS2, protect neurons against excitotoxicity, here as NMDA treatment or glutamate, or following the apoptosis. Also protect synapses against the MDA treatment. Here, neurons treated with MDA lost the synaptic, the dendritic spine, and neurons co-treated with imatinib or GEMS2 inhibitor, CBL inhibitor, were protected, and also were protected when we analyze the levels of NDL receptor phosphorylated in NR2B subunit. Then CBL is involved in the damage induced by glutamate or MDA. But we go to in vivo models. For in vivo models, we use it wild type and null CBL, mice are null for CBL in the brain and induced status epileptico by injection with pilocarpine. Here I show you that the animals that wild-type animals are injected with pilocarpine, all animals have seizures. However, only a fraction of the animals are null for CBL have seizures, and the animals that present seizures take longer time to reach the seizures, and more animals have seizures survival after the treatment. Then the CBL is involved in the mechanism that are altered during the seizure as a model of epilepsy. But as you remember, probably remember, CBL is involved, has been described, is involved in cytoskeleton remodelation and gene expression. Then we asked if CBL is involved in this function in healthy neuron. Interestingly, we observed that CBL signaling is involved and is required for the BDNF-induced arborization in hippocampal neurons. In hippocampal neurons, BDNF induced the growth of dendritic tree and its branching by binding at its receptor tract B, and we observed that BDNF induced tract B activation, but also induced the activation of CBL. And this activation also was associated with the tract B and CBL interaction, here followed by immunoprecipitation. But more important, BDNF induced the arborization, the dendritic arborization in hippocampal neurons, yes, and increase in size and branching of dendrites. But when the neurons were treated with BDNF, animatinib, or GM2, or when we use it, neurons are null for CBL, this effect of BDNF over dendritic tree growth was prevented. With this in mind, we went to evaluate the cognitive performance of young mice that are null for CBL. Interestingly, we found that the evaluation of CBL was associated with a better learning and memory. Here, evaluated by 3 cognitive tests: Barnes maze, Morris Water maze, and novel object recognition. You can see here that the animals are null for CBL, showed better performance in memory acquisition and consolidation that wild-type animals. Previously, evaluating the roles or function of CBL in the neurodegenerative disease, we described that the CBL is involved and participate in the neuronal gene repression through modulate the stabilization and levels of HDAC2 protein. With this in mind, we evaluated the gene expression of a wild-type mice in basal and training conditions, and CBL null mice, basal and training animals. We isolate the hippocampus at day 2 of training and performed a RNA-seq analysis, and we found more around 300 genes that are regulated by the cognitive training and the presence or absence of CBL gene. We observed that the gene, using ontology analysis, we observed that the genes that are upregulated when CBL is absent are mainly, and the animals are trained, the genes are related with spin morphogenesis and dendritic spin development and cytoskeleton. And our results suggest that CBL restricts the expression of genes involved in cytoskeleton remodeling during learning. This result was consistent with the observation that the animals that are null for CBL in the brain and are trained present more dendritic spines than the wild-type mice after the training. And also, if you have neurons are null for CBL and stimulate with chemical LTP, you observe an increase in effective polymerization. Altogether, our results sustain that the CBL activity and probably also ABL2 activity are involved in ORK for signaling for synapses remodeling, and these kinases are in a crossroad between synapses healthy function and damage, and its activation and localization give us a fine tuning between healthy synaptic remodeling and neuronal and synaptic collapse. Finally, I want to thank to the members of my laboratory that are working hard in these results. I show you Daniela Gutierrez, Gabriela Larenas, Álvaro Cáceres, Lina Vargas, Nancy Leal, Pablo Contreras, Adrián González, and the previous member labs as Ignacio Cancino, Marcelo González. Thanks also the members of Silvana Sanlungo Lab and our collaborators and the funding. Thank you. Bye.
Okay, thank you very much, Alejandra. Very nice talk. Have a huge amount of results. We already have a question that was made by Gonzalo Olivares. He asked, how can be explained that different stimulus activates CAB8 and that this can be related to different disease? That it depends on the targets that are related?
Well, in fact, we don't have the final explanation. Sorry, because you can repeat the question because I have.
That's okay.
We don't have a clear answer about this because CBL is a very complex kinase because it has different subcellular localization, have function in the nucleus, also have function at cytoskeleton and in membrane, and have function in ER and other and probably lysosome. And this activation also is related with different target protein downstream. Then our idea is this kinase is involved in different stress or signal that modulate the response to damage. Indeed, also some results could be related with the level of CABL activation. CABL activation have different step for its activation, have a conformational change and phosphorylation that are go in increasing the activation of this tyrosine kinase. I don't know if I answered the question.
Okay. There's another question made by Paola Hager that say that Soto KO of CABL by itself has effects on urbanization. How you can explain that?
Well, we are really integrated because we have results that show the CBL functions required for synaptic and neuronal process remodeling, and is activated by BDNF and is required for this pro-survivor or good signaling, but also is involved in dendritic loss. We think this is a balance, and the function of the CBL is required for the this ensemble of the synaptic and neuronal process structure for create new synapses or dendrites. I think this is the...
Okay, that's a combination, you mean. So there are a few more questions that we will keep them for the last, at the end, for the discussion. Okay. Now we are going to the third speaker. Our third speaker is Dr. Susana Castro from Mexico, DF. Susana is a bachelor in basic biomedical research, master in biotechnology. She also made a PhD at the National University of Autónoma from Mexico. Since 2013, she is a senior research B at the Neuroscience Division of Neurodevelopment and Physiology Department at the Instituto de Fisiología Celular, UNAM. Her research is focused on defining the molecular bases that regulate the autophagy and its influence on the development and aging of the nervous system, as well as the stability of genome in mammals. The title of her talk is Autophagy Dysfunction Contributes to Neuronal Senescence. Let's see that.
Hi, good day. Thank you very much for your kind invitation. I would like to start by introducing the members of my lab. They are grad and undergrad students, and together we study the molecular and cellular processes that are necessary for proper embryonic development, but later in life contribute to aging, such as cellular senescence. And we use as a model mice, rat, neckmole rat, and human cells. Today, I will present our results that led us to conclude that autophagy dysfunction contributes to neuronal senescence. I'm going to start by defining aging as the molecular, cellular, and organismal changes that occur over time that increases the likelihood to die. And this is a nice review that summarizes the main hallmarks of aging, which are those changes that occur in several species. And what is very interesting is that not all species age at the same age. For example, the neckmole rat, it's a rodent that lives like up to 30 years, but do not develop these age-related symptoms and diseases. They do not develop cancer, neurodegeneration, osteoporosis, etc. And the mortality hazard does not increase with age. And for example, as opposed to humans or the horse. So we are interested in studying at the cellular and molecular level what are differences in the cells of these different organisms. And among the different processes that can change is autophagy, and we are particularly interested in it because at least 8 out of the 9 hallmarks of aging are consequences of autophagy dysfunction, demonstrated in multiple publications in different species. And with age, the efficiency of autophagy is diminished in the species that age, but not, for example, in a neck mole rat. If you compare the efficiency of autophagy or the functionality of autophagy in an organism that is 25 years old, is as efficient as a young animal. And the different mechanisms of autophagy, we focus on macroautophagy, and I would like just to highlight a few features, in particular the function of LC3 protein, that is a protein that is anchored to the phagophore, that is a precursor membrane that will elongate surrounding the cargo, and it will be directed, the direction of the elongation, by the interaction directly with the cargo through an autophagic receptor, that is a protein that has the ability to bind both the LC3 and with the cargo that has been labeled, for example, with ubiquitin. And this gives rise to this specialized vesicle called autophagosome that eventually fuses with the lysosome to allow the digestion of the cargo together with LC3 and the autophagic receptor. So if this process gets interrupted in some steps, it leads to autophagic flux impairment. So what we mean with that is that the degradation is interrupted, and some of these precursors accumulate inside the cells. And that is what happens in aging. And what is give hope is that the pharmacological or genetic interventions that extend longevity in several organisms, what they do is that they stimulate autophagy. And on the contrary, interventions that inhibit autophagy reduce longevity. And we focus particularly in the brain because, at least in Drosophila and C. elegans, it has been shown that increasing autophagy only in neurons is enough to extend longevity. And the opposite, reducing autophagy in the brain in mice, causes neurodegeneration. So the brain seems to coordinate organismal aging. And among the different hallmarks of aging, we focus on cellular senescence because it has a pleiotropic effect. And it was so because the senescent cells have a particular phenotype, and the most important feature is that they secrete different molecules that alters the tissue where they reside. They secrete cytokines, growth factors, metalloproteinases, etc., that collectively are known as the senescent-associated secretory phenotype. Another feature important of cellular senescence is that the cells no longer divide. They are in a perturbable cell cycle arrest. These cells are resistant to apoptotic and mitotic stimuli as opposed to quiescent cells. And they also have other features, like, for example, they have an increased activity of a beta-galactosidase-like enzyme. They accumulate lipofuscin because the lysosomes are not good at digesting anymore. They overexpress cell cycle inhibitors P21 and P16, and they have also changes in the nuclear morphology, in the nuclear envelope in particular. Lamin B1 and its receptor are degraded, and this changes the structure of the chromatin that leads to changes in the pattern of gene expression. Lamin A gets stabilized, and they have this clearly nuclear aberrant morphology. They also have a sustained DNA damage response that can be identified by locating a dephosphorylation of this variant of histone. And they are harmful, senescent cells, when they are persistent in the tissue because they alter the tissue architecture, they induce paracrine senescence, spreading the phenotype, and they induce chronic inflammation. So we can hypothesize that these same things could be happening in the brain. Cellular senescence was originally described for mitotic cells, so it was even like considered that, like out of the imagination, that post-mitotic cells could develop the senescent phenotype. But the features that we observe during physiological aging and in neurodegeneration could be a consequence of senescent cells. So we look for senescent cells in the brain, and we found them. In the cortex in rats and in mice, in cortex and the hippocampus, we found cells with higher beta-galactosidase activity. They accumulate lipofuscin. They overexpress P21 and P16, which was, surprising because neurons are already post-mitotic, so there is no need to inhibit further the cell cycle. So that leads us to think that P21 and P16 may be having additional activities that contribute to establish the senescent phenotype. The neurons have also nuclear alterations. They have a DNA damage response, persistent activation, and most important, they also secrete SASP molecules. They overexpress this transcription factor, GAT4, which transcribes several of the SASP genes. And we found that they, for example, overproduce IL-6. And so in order to study the molecular basis for the neuronal senescence establishment and maintenance, we developed an in vitro model of neuronal senescence. And here we show you that just culturing primary cortical cells for 26 days, they eat without any insult. They develop the senescent like phenotype. They have increased beta-galactosidase activity. They accumulate lipofuscin. They have nuclear alterations. They have P21 and P16 expression. They have DNA damage response, sustained activation, and most important, they also secrete the SASP again. They overexpress the AG4 transcription factor, and in conditioned media from 26 days culture of cortical neurons, we found that they also secrete these cytokines. In particular, it's important MCP1 because it was shown that it induces paracrine senescence, and we demonstrate in conditioned media that also on neuronal senescence secrete MCP1, well, secrete this bunch of molecules that induce paracrine senescence in non-neuronal cells. So mechanistically, we thought that the link between autophagy dysfunction with senescence could be the accumulation of GAT4 and P16 because these are autophagy substrates. So if the autophagy degradation is impaired, the autophagy function is impaired, it leads the accumulation of these two proteins, which we observe. So we monitor autophagy by detecting autophagocytes with this compound, SALT-ID. We can induce it with 3 halogens, we can inhibit it with spouting or with chloroquine. So the first step was to study if indeed the neurons in the brain had autophagy, autophagic flux impaired, and indeed here there is an accumulation of LC3 and P62 in the neurons, and the same happens in our in vitro model of neuronal senescence. There is an increase and accumulation of autophagosomes. LC3 and P62 are not degraded. That can be confirmed also by Western blot. And by immunogold, we detected also the accumulation of autophagosomes that are no longer being able to fuse with the lysosomes. And the autophagic flux is indeed impaired because adding chloroquine that inhibits the fusion of autophagosomes with lysosomes does not increase further the LC3 or P62. And we also demonstrate that cells that have the autophagy dysfunction, like here, like by the accumulation of LC3, have also senescent features like the nuclear abnormality. So the next step was to study if modulating autophagy could increase or diminish the proportion of neuronal senescence at these different windows. We either induce or inhibit autophagy, and we observe that indeed inhibiting autophagy with spouting the second and third week of culture, we have more senescent neurons, and the opposite, inducing autophagy reduces neuronal senescence, and it was confirmed looking at other senescent features like nuclear morphology. So the next question was, why is autophagy impaired with aging? And here we put together several information generated by others. A colleague in Mexico, Dr. Guillermo Lanuza, had discovered that in premature aging cells, he demonstrated that the fibroblasts become senescent and that they have overexpression of CREM1, which is exporting that translocates that exports nuclear cytoplasm into the nuclear protein, into the cytoplasm. So having an exacerbated activity of CREM1 leads to a reduction of nuclear proteins and an increase of cytoplasmic proteins. And treating the cells with leptomycin, that is an inhibitor of CREM1, restores the nuclear localization and reduces the cellular senescence of fibroblasts from premature aging. And on the other hand, there was another report in which they show that inhibiting exporting 1 from 1 increases the nuclear localization of the transcription factor ED, which is a master gene for the expression of autophagy genes and lysosomal genes. So it's necessary for the biogenesis of lysosomes. And in doing so, it is increased autophagy and it is enhanced longevity in C. elegans, and there is also another report that in Drosophila happens the same. So Elisa asked the question whether autophagy will be impaired in the aging brain due to an increase in CREM1 expression. And so she did an analysis in these hippocampal regions and the cortex, cerebral cortex, and indeed, CREM-A is overexpressed in aging. This is the icon for this is in mice. And I'm sorry. And concomitant with an increase in CREM1 expression, there is exclusion of TFEB from the nucleus and also of STAT3, that is a protein that when it is located in the cytoplasm, it also inhibits autophagy. And indeed, the autophagy flux is impaired. The same cells that have increased CREM1 expression have the P62, the autophagic receptors accumulated, and they also have senescent features as the expression of P16, and it happens in neurons. The same happens in our in vitro model. The neurons in vitro also accumulate CREM1 expression, which correlates with the appearance of senescent features such as P16 expression, changes in the nuclear structure, degradation of lamin B1, and an exclusion of TFEB from the nucleus. And more interestingly is that when we inhibit CREM1 with leptomycin, the nuclear localization of TFEB is restored. And with this restoration, also the autophagic flux is restored. Here we can see how P62 is accumulated in neuron culture for 26 days, as we have shown you. And when we add leptomycin, P62 is once again degraded, and the same happens with LC3. And what is more interesting is that also senescent features are reduced with CREM1 inhibition. Here you can see here we are detecting beta-galactosidase activity. You can see how it is increased in neurons at 26 days, and with leptomycin it is reduced. So in conclusion, the pharmacological inhibition of CREM1 restores autophagic flux and reduces neuronal senescence. And so we propose, so we think now that interventions that could restore the autophagic flux in vivo could prevent or even restore neuronal senescence and age-related cognitive dysfunction. And with this, I would like to end, acknowledging previous members of the lab, collaborators that I have, and the financial support that I have received. Thank you very much.
Thank you very much, Susana, for your presentation. Very nice results. Let's see if we have any question posted. And so one question I have, it's if CRM1 increase the exportation of neuronal proteins, nuclear proteins, sorry, how this function is altered in the autophagic flux?
Yeah, so the idea is that the exacerbated activity of CREM1 leads to the export of transcription factors necessary for autophagy induction. So if they are located in the cytoplasm, like TFEB, if it is located in the cytoplasm, it can no longer transcribe autophagy genes. And this other protein, like STAT3, it inhibits autophagy when it is located in the cytoplasm. So it will be a combination of this activity of these molecules that will impair the autophagy.
Okay, we have another question from Jaime Fornaguera. Did you study the proposed effects of autophagy deficiency by older animals using behavioral tests related with the motor and cognitive performance?
Yeah, no, we haven't tested yet in vivo. This is our current studies. We demonstrated, so have just characterized what happens in the brain, but now the modulation of the inhibition of the CREM1 activity in vivo is currently in progress. Yeah, and we will do definitely the cognitive. Yeah.
I have a question.
Sorry?
I have a question. Can I ask a question right here? Okay. So very nice talk. I'm wondering, considering the end of your talk, from a therapeutic perspective, do you imagine it could be doable to try to enhance autophagy specifically in affected neurons? I mean, because if we think about something like general induction of autophagy could be detrimental in some way. I don't know actually, but are you planning to test a more specific neuronal type or in a single nucleus of the brain to try to induce it?
Yeah, we think, and it has been observed that autophagy, when it works properly, it is, I mean, you cannot like over-activate it to have, you know, like a negative effect in non-affected cells. Actually, we try to induce autophagy in vivo, like in young animals, and I mean, there is no way because it is very well regulated. And we have been trying to induce autophagy by several non-invasive methods. Like there are in vivo, there are a lot of information in animals, like fasting, for example. Intermittent fasting will induce autophagy, or exercise induces autophagy, even in the brain. And yeah, so we think that it could be preventing the development of the senescent phenotype. And so yeah, we are basically looking for like more non-invasive and easily translatable approaches to prevent the onset of this. Yeah, thanks.
That's great. Thank you, Susana. Then we continue with the discussion later. So now I'm happy to introduce our fourth speaker, Dr. Mychael Lourenco from Rio de Janeiro, Brazil. Mychael is a biologist and made his PhD at the Federal University of Rio de Janeiro. He's currently an assistant professor of neuroscience and a group leader at the Laboratory of Neurodegenerative Diseases, part of the Institute of Medical Biochemistry Leopoldo de Meis at the same university. His research focuses on the understanding of molecular mechanisms of neurological diseases, with emphasis on Alzheimer's disease and depression. His lab studies the metabolic and molecular changes related to cognitive impairment found in patients with AD, with the proposed to correct these metabolic disorders through pharmacological approaches or physical exercise. The title of his talk is Mechanisms of Neuronal Dysfunction and Neuroprotection in Alzheimer's Disease. Thank you, Mychael.
Thank you for the kind invitation to talk at this FALAN symposium. I'm very honored to be here talking about research that we have developed in our laboratory over the past few years. So I'll be talking about some mechanisms of neuronal dysfunction and also strategies of neuroprotection in the context of Alzheimer's disease. Well, as you know, Alzheimer's disease has memory loss as its main symptom. And unfortunately, there's no single mechanism that could explain the pathology of Alzheimer's disease, though we know that age is the major risk factor for developing dementia and Alzheimer's disease. And unfortunately, again, we do not have any effective diagnosis or therapy that halts or even reverses memory loss in patients. And if I were to tell you about what happens in terms of progression of Alzheimer's disease, one of the first or earliest events in Alzheimer's disease is the accumulation of amyloid beta peptides. Then there's synaptic dysfunction, tau-mediated injury, and then the symptoms occur. So there are several molecular phenomena that happen before symptoms occur in patients. So there's an urgent need to try to understand what happens before disease takes place, before the symptoms take place in the patients. So what we know in terms of Alzheimer's disease is that there's overt accumulation of amyloid plaques, which are mostly composed of amyloid beta peptides derived from a proteolytic cleavage in the neuronal membrane. And amyloid beta has the tendency or the trend to aggregate and forms oligomers too, and also fibrillar structures that are insoluble. While the amyloid plaques have been largely regarded as important features of Alzheimer's disease, we now know that A-beta oligomers, which are soluble and they are able to diffuse throughout the brain, they are considered important toxins in the context of Alzheimer's disease. They bind specifically to synaptic contacts, to synapse receptors, and they also trigger oxidative stress, tau phosphorylation, and also they cause synapse failure. And the greatest question that we should ask is what leads, in fact, to synapse and memory failure in the context of Alzheimer's disease? And once we know that, how can we use that information in order to translate into effective therapeutics? That's what we have tried to do over the past years in the laboratory. And we've also got some clues from lifestyle and life impacts in the context of risk of Alzheimer's disease. We know from several studies that insulin resistance, type 2 diabetes, obesity, all metabolic derangements and inflammation are tightly connected to the risk of developing Alzheimer's disease. And we know that physical exercise is a non-pharmacological approach that is able to bring benefits to not only to our body, but also to the brain. So it improves memory in healthy people, improves attention, and then also brings positive effects for some sort of neurological disease. And in terms of Alzheimer's disease, we know that walking regularly is able to stabilize cognitive functioning. That's not the cure, that's not the reversal of the disease, but it also promotes some sort of benefits for Alzheimer's disease patients. And we also know that people who regularly exercise throughout life, they have a reduced chance of developing Alzheimer's disease, though we know little about the potential mechanisms that are neuroprotective and they're induced by physical exercise. That's something that we try to address. And we focused on a protein, a hormone called FNC5/irisin, mostly known as irisin. It was described almost 10 years ago, and it is a fragment of a precursor protein called FNC5. And irisin is induced by physical exercise. It's produced by the muscle, skeletal muscle mostly, and released into circulation after exercise sessions. So this is the precursor protein that is cleaved and released into circulation once we exercise. And the lab of Bruce Spiegelman at Harvard, they described that subjects that exercise have higher levels of irisin in their plasma. And when they administer irisin to obese mice, they have improved glucose tolerance. So it brings important metabolic effects in the body. And there's also some evidence that irisin is produced in the brain and also that it may regulate some neuroprotective pathways in the brains of mice and humans. So we asked basically three questions here first. The first thing that we like to address is whether irisin levels are altered in Alzheimer's disease. And so we had an opportunity to collaborate with Joe Sambra at the University of Kentucky at that time to investigate the levels of irisin in the hippocampi of Alzheimer's disease patients with early Alzheimer's disease and patients with late Alzheimer's disease. And we found that patients with late Alzheimer's disease, they had reduced levels of irisin as compared to early AD or to control patients. And collaborating with the Dora Institute here in Rio de Janeiro, we observed that there was no change in the plasma levels of irisin in dementia, nor in MCI patients. So here we observed that plasma irisin is preserved throughout the disease, either early, let's say in MCI, mild cognitive impairment patients, or in Alzheimer's disease, but also in Lewy body dementia. So there was no change in the plasma level of irisin. But when we looked at the CSF the cerebrospinal fluid of Alzheimer's disease patients, we saw a clear reduction in the levels of irisin in the CSF. And also, it also happens with Lewy body dementia. So it's not specific to Alzheimer's disease, but also to another form of dementia. And you can see here there's a considerable variation in controls, and that's sort of expected as people have different habits, people exercise more or less, and they have different activity profiles. But when they have Alzheimer's disease, levels of irisin go down. And in a follow-up paper that was published last year, we could also observe that irisin levels in the CSF correlate with memory and also with beta amyloid levels in the CSF too. So that shows some sort of relevance, potential relevance with memory function and also Alzheimer's disease progression. And it also correlates with an important neurotrophin, which is BDNF in the CSF too. And you might remember that I showed you that A-beta oligomers are important neurotoxins in mice and also in humans. And when we looked at the brains, at the hippocampi of A-beta oligomer injected mice, we also could observe or mimic irisin reductions. So that sort of shows that the elevation in A-beta oligomers in the brain might be causally linked to irisin reduction in the hippocampus. So is there any relevance of this reduced FNC5 irisin in the brain for memory? So the next thing that we decided to do is to silence irisin expression in the brain by injecting lentiviral vectors harboring shRNAs. And when we did that, we got a good level of downregulation, about 60% in terms of mRNA and also in terms of proteins, using two different constructs. And what we observed is that these mice have impaired memory when exposed to a novel object recognition task. So if they are silenced for irisin, they cannot distinguish between a familiar and a novel object in this memory task. That doesn't happen with control. Controls perform normally. And when we looked at the synaptic plasticity in their brains, we observed that whereas both mice are able to form or to have increased excitatory potential, mice that are silenced for irisin, they cannot maintain this throughout time. So that's important because it shows potentially a problem with memory consolidation. And that said, something that we need to address here is whether irisin could be neuroprotective in terms of Alzheimer's disease. And we did that by first exposing hippocampal slices to irisin and A-beta oligomers. And while we saw something that's very well known in the literature, that A-beta oligomers are able to impair synaptic plasticity, this is long-term potentiation, when irisin is present, that doesn't happen. So it means that irisin is able to protect or to correct A-beta oligomer-induced synaptic plasticity impairments. And when again we did novel object recognition with mice, but now injected with A-beta oligomers, we also know that they do not distinguish between novel and familiar object, meaning that they have impaired memory. But when they were injected intrahippocampally with irisin, most of the mice are able to discriminate these objects, showing that irisin is able to correct memory impairments induced by amyloid beta. And we also did similar experiments in a transgenic mouse model of Alzheimer's disease, which is the APP-PS1. And here we used an adenoviral vector to overexpress FNC5, which is the precursor of irisin. And basically we had same memory benefits. Here we have a radio arm water maze task in which control APP-PS1 mice, control transgenic mice, they have impaired memory, so they have more errors. But when they overexpress irisin, they just perform as wild-type mice. So they have memory preservation or restoration induced by irisin in this model. But there's a question that's an important question that I didn't mention so far, is whether irising mediates the brain benefits of exercise in Alzheimer's disease. So we did a kind of sort of complicated experiment here, but what you have to look here is that when mice exercise, they are protected against a beta oligomer-induced memory impairment. But when they received an injection of an antibody to block circulating irising, most of the mice cannot perform well in this memory task, in this novel object recognition. So that means that irising, at least in part, mediates the beneficial effects of physical exercise in the brain in the context of Alzheimer's disease. And that's all published in a paper that we published in 2019. And that was very well received because it correlates with human data showing that lifestyle changes are important to prevent Alzheimer's disease. So lifestyle changes, which include practicing more exercise, having more physical activity, is able to protect against the risk of Alzheimer's disease, even if you are at higher risk of Alzheimer's disease. So that's really important because it shows that there's something we can do in our lives to prevent Alzheimer's disease. And what is this combination of lifestyle changes? So not smoking, exercising, controlling nutrition, controlling what we eat, controlling what we drink, and also trying to stimulate the brain with pro-cognitive activities. And in the last 2 minutes that I have here, I want also to show some work that we've been doing with the molecular mechanisms of memory loss in Alzheimer's disease. So several years ago, we published a paper in Cell Metabolism showing that there's cellular stress and neuronal stress in the brains of Alzheimer's models, and that this is responsible for inducing memory impairment. There was right in time, just in time, there was a paper by the Eric Plan Lab showing basically the same things with another approaches and with another upstream targets. But basically, what we showed is that A-beta oligomers induce an inflammatory response that activates cellular stress and that impairs the initiation of protein synthesis, leading to synapse loss and to memory impairments. And there were some advances in the field, and there was a pharmacological compound that is known as ISRIB, that was developed by Peter Walter Lab in San Francisco a couple of years ago. And we decided to test whether this compound, ISRIB, would prevent or would block the deleterious effects of cellular stress in the context of Alzheimer's disease. And we published a paper earlier this year. I was fortunate to collaborate with the group of Sergio Ferreira, and we were happy that it was chosen as the cover of Science Signaling in February, and which we show that ISRIB is able to rescue cognitive impairment induced by amyloid beta. So you see here a novel object recognition test. This is the discrimination index. Amyloid beta injected mice, they perform poorer. And then when they are injected with ISRIB, most of the mice perform better. So that shows that this integrated stress response pathway is important in the context of amyloid beta toxicity. So that stimulates future studies targeting this pathway that we described a few years ago. Well, in conclusions, I want to say that it looks like defective hormonal signaling is present in its brains. Exercise is neuroprotective. Irising might be an important connection in this exercise-induced neuroprotection. And perhaps correcting brain hormonal signaling and protein synthesis would be important strategies in the context of Alzheimer's disease. I want to thank my several collaborators, funding agencies, and I'll be more than happy to discuss with you any questions that you might have. This is the large group that I'm part of. These are my students. Thank you very much.
Thank you, Michael, for your presentation. Very nice results. So we do have a question from Gonzalo Olivares. What is the source of irising in the CSF?
Okay, so estoy muy contento por hablar en FALAN, estoy hablando español porque creo que muchas de las personas son de Latinoamérica, but I'll change to English as someone may not understand it. So we believe it to be both. There's local production of irisin in the brain, so it may be an important source of CSF irisin. But we also believe that there is irisin coming from the periphery, coming from the muscle, then going to circulation, and then being transferred to the CSF. There's a recent paper in Nature Metabolism by a Harvard group sort of indicating that irisin that's produced in the muscle or in the periphery also goes into the brain. So I think that CSF pool of irisin could be from both sources.
Okay, I don't know if there's another question. So, do you know if there's any receptor for this hormone in the neurons?
We haven't looked at that by ourselves, but there are some descriptions, including this recent paper that I just mentioned, saying that an integrin, alpha V, beta 5 integrin, could be one of the receptors for irisin. So that was just recently described a few months ago.
Okay. So let's start the round of questions for all the speakers. So to do final remarks. Let's see, we do have a preliminary question for Elena.
Yes, I think someone in the chat dropped a question
Yes.
that I need to answer. So, but I cannot find it now. It's so long.
I have it here. So Diana Jerusaliski asked, is there any other intracellular abnormal protein accumulation at this time detectable for abnormal protein accumulation, and whether is the excess of CR protein located?
Okay.
That was one place.
The first one, yes, of course, there are many other proteins that can accumulate inside the cells, but in the case of our model and in tauopathies in general, we know that the main protein in neurofibrillary tangles is tau protein, and it is hyperphosphorylated. And there are also, this is quite new, some reports showing that within this tau accumulation, we can find some no-coding RNAs. And this goes linked to another question that comes later, and I remember now. And the second part of the question of Diana was where it is located. So, yes. Well, it's mainly, as in all tauopathies, the accumulation of tangles is in somatic. And this is a key feature of tau dysfunction, because we know tau is mainly in the axon, because although tau is exerting some function in the soma as well, we need tau to go to the axon to regulate microtubule dynamics. And when something goes wrong with tau metabolism, it stays in the soma, and we find these inclusions in somatic inclusions either in humans and in our models. And someone else asked a question that I tried to answer in the chat as well, and it comes with this as well. It was related to long no-coding RNAs that might be regulating splicing. So, so far, there is nothing published specifically for splicing regulation of MAPT by long no-coding RNAs. However, there is an article that was probably the person that asked this question was aware of this, that came up in, I think it was Nature this month or last month, showing that long no-coding RNAs could regulate MAPT expression. So basically by binding to promoters. So it's at the level of DNA, not RNA. But okay, I think it's a growing field, and something will come up soon with that. So I think I'm fine, but I have another question that I answered in the chat. Sorry, it was about other modifications that could affect isoforms post-translationally. And the answer is yes, because we have many phosphorylation sites in the microtubule binding domains. So if we have 3 repeats or 4 repeats, of course, that will affect the number of phosphorylated epitopes, and that will also affect binding to microtubules. So the answer is yes. The post-translational modifications, specifically phosphorylation, will also affect the binding of tau to microtubules.
That's
Sorry,
great.
I was
Thank
quite
you.
fast.
No, it's okay. So let's go to a few questions that are left for Alejandra. So the CABL activation by NMDA and glutamate went down at higher doses. Why is that? The answer about your results. You have to activate your microphone.
Well, we observed the activation of CBL after MDA or glutamate treatment, and I don't understand the question because it's a comment, not a question.
No, they say why is that the CBL activation by NMDA decline at the higher doses? It's reduced at higher doses. Do you have an explanation for that?
Well, we saw the CBL is induced apoptosis signaling and its activation by excitotoxicity. We don't have the mechanism involved why MDA or glutamate induce CBL activation, but the blocking of this CBL activation reduce the apoptotic signaling and the cytoskeleton alteration. There are some reports that indicate that the CBL is associated with MDA receptor in postsynaptic yes, and could phosphorylate some subunit, but there are not really a mechanism linking the activation of MDA directly to CBL that describe it for now.
Okay.
I don't know if
there's a comment from Paola Hager. It is also impressive that the inhibition of kinase ABL avoid the toxicity of 100 micromolar NMDA.
Yeah, it's about the doses of CBL of MDA. Yes, we have a result with 150 micromolar MDA and also with a lower concentration. But it's a comment about the high doses. I don't know. Okay. I don't know what the question is.
Yes, okay. So another question from Veronica Baez is if it is possible that the ABL function as a scaffolding protein also.
It's not, well, in ER was described the CABU could be associated with some protein are participate in the scaffolding of ER-alpha receptor in UBR response. And also CABL is associated with cytoskeleton and is associated with PSD95 we described some years ago. Indeed, CABL is not a scaffolding protein, but interact with scaffolding protein in different contexts, in the cell membrane context, and in other membranes such as ER and lysosomal membranes. It's not a scaffolding, but it's probably is modulating scaffolding protein and is remodeling this complex.
Okay, thank you. So let's move to another pending comment that there was a post for Susana. Jaime Fornaguera says that the regulation of autophagy is complex and might be promoting autophagy. You can affect other processes that impair physiological functions. So do you want to add some extra comment on that?
Yes, I would like to explain a little bit better. So what we observed in senescence in neurons, and also that in other people have described in neurodegeneration, is that the autophagic flux is impaired. That means the fusion of autophagocytes with the lysosomes is impaired. So the lysosomes are dysfunctional. And under this situation, it's true that if you induce the activation of autophagy, I mean the production of more autophagocytes, you will make it worse for the neurons because they cannot go to the further step. So what we think that we found by increasing the activity of TFEV2 by reducing the activity of CRAM1 that keeps it in the cytoplasm is that we can restore the lysosomal biogenesis, and we can restore the autophagic flux. So it is true that it's complicated. It's not enough to just induce the activation of autophagy in general, because it may make it worse for the cells that it has already impaired if you just induce the formation of new autophagocytes, but it is just to restore the autophagic flux. And it is true that autophagy has a lot of functions. It's an integrator of the metabolism, actually coordinates anabolism with catabolism. And so that is why we think that it is really important to try to prevent or to restore the autophagic flux when it gets impaired with aging. And yeah, it's true that it can be sometimes seen autophagy as the yin or yang, like too much is perhaps not good or improperly activated. And yeah, it is true.
Okay, thank you. Let's see, there are some other questions from Michael. Sandra Zarate asked you if the experiments were done with both male and female mice. If you don't, would you expect to have any difference on irising levels between the sexes?
That's a very good question. We had several experimental models, and we had both males and females, and we saw no difference in terms of neuroprotection. So irising appears to be effective in both. I know that there is another group looking at specifically, I think, tau phosphorylation and some other stress-related cognitive dysfunction, and they preferentially see effects of irising in females, but we saw a good result for both sexes. And actually, we have not tested in terms of aging in mice, but we know that there is an increase in irising levels in humans across aging. So that may be part of a protective response. We cannot say much about what it means, but it looks like normal people with preserved cognition, they have increased levels of irising as they age in the brain, in the CSF. But that doesn't happen with people with Alzheimer's disease or Lewy body dementia. Okay.
So another question from Cecilia Conde is if the irising is expressed in muscle cells, how has it a known role on other pathologies like Huntington or Parkinson disease with protein aggregates? Do you know something about that?
Yeah, that's a good question. We actually don't know. It's important to study that because Huntington and Parkinson's are diseases with muscle and motor impairments, but that's something we don't know yet. But what we know in terms of brain is that irising is preferentially expressed in the hippocampus, and there's something in the cortex, but I'm not sure about specifically the motor cortex or even peripheral neurons that control movements.
Okay, there's another question in this sense. What type of exercise increase more irisin in the body or in the brain? Claudio Pinto says that.
We preferentially investigated aerobic exercise, not strength ones, but we know that there are reports that strength exercise also increases iris in humans, but we investigated swimming and running. So, and mostly swimming in our paper, but there is evidence that running does it as well. Okay,
so there's another comment for Elena Avale from Natalia Contreras. She said that the DNA epigenetics marks a long coding RNA and transcription on co-transcriptional, so alternative splicing could be regulated by long coding. No coding. I don't understand the cycle RNA. What do you think?
Yeah, I was trying to answer in the chat because I thought there would be no more time for discussion. But yes, Natalia is pointing towards a very important. I think it's a very interesting topic because it is well described that many transcripts, alternative splicing is regulated by this long non-coding RNA. So I'm sure that it will come up soon. Some studies specifically related to tauopathies, right? Because something that I mentioned in my introduction, probably I did not have enough time to go further on that, is that I don't know if I can share now, but if not, I can just talk about it. Is that the regulation of alternative splicing in the tau, in the MAPT transcript, is not only regulated by cis-actin sequences, which are around in the boundaries of the exon 10 in particular in this case, but there are many factors that has proteins, splicing factors that will lead to interact with the transcript as in any splicing regulation. And it is known that these splicing factors are in turn regulated by, for example, microRNAs. So the level of a given factor that will bind to the MAPT transcript will be regulated in a given neuron in a given moment of development or any reason that the plethora of factors that could affect that. But it is regulated, for example, by microRNAs. So it's quite complex. The regulation of exon 10 splicing is very complex. So please do not think that the only reason why isoforms imbalance is could occur is due to mutations in the MAPT gene. There are many other factors, either genetic that could be affecting the splice factors, or epigenetics, or environmental, or whatever. So there are many examples of this. We do not have time to go in deep with this, but just to mention one, I always like to give this example, is in a French island, in Guadalupe Island. It was described that there were many patients developing a 4R tauopathy, particularly PSP, progressive supranuclear palsy. And when they found out that they had a huge increase in 4R tau in the brain, the researchers, French researchers, went, and the first thing they analyzed was a sequence in the MAPT gene of those patients and the pedigrees, and nothing was found. And to make a long story short, at the end of the study, they found out that exon 10 splicing was affected by a substance that these people used to take in a tea, in a typical tea that they take in Guadeloupe, which is enriched in anonacin. So as we have mate, they have this other drink, and that was affecting tau splicing. So it's very complex. So just to open up the discussion and do not have in mind, maybe I was so specific to mutations in my talk, but there are many other factors affecting tau splicing.
That's very interesting, Elena. So let's post another question for Alejandra. Claudia Espinosa Garcia is asking that it is worth to explore the role of ABL in microglia as well. What do you think about it?
Sorry. Yes, we want to explore the role of CBL in microglia and glia astrocytes in general, but we don't do this yet. We didn't do this yet, but we have some results that show the CBL is not expressed in high level in glia. The level of the expression and the activation of the CBL in glia is not important. However, we have a result in immune cells, in lymphocytes, and we have a similar result we have in neurons, we have in lymphocyte. The CBL activated in the cytoplasm, when we activate CBL in the cytoplasm associated to cytoskeleton, we have survival and cytoskeleton remodeling. But when the CBL is activated and go to the nucleus, induce apoptosis and down gene expression repression, repression of genes. Then in immune cells and neurons, we have this different, the location of CBL is very important for the downstream signaling, but we don't understand now which modulate this if the CBL signal go to the nucleus or go to remodulate cytoskeleton.
Okay, that's okay. So I don't know if any of the speakers want to add any other comments, but I think we are almost too late for lunch. We do have to make a break. I made a forum for discussion at the group of FALAN 2021 meeting. You will find it to further discuss these topics. So I would like to invite all the audience to participate. And I already have some extra questions to move it also. So one more time, thank you very much for Alejandra, Susana, Mychael, and Elena. That was a very nice symposium. So I think we are done. So after lunch at 2 p.m. the sessions will start again. Okay. See you soon.
See you.
Thank you very much.
Good afternoon to all participants of FALAN 2021, this networking symposium that we are having. It is a pleasure this afternoon to have Paola, Christian, and Marcela. I will introduce their talks and we're gonna get to know the FALAN, but not only we're gonna get to know FALAN, but also a map of neuroscience in Argentina and some interesting general information about science that Christian is going to show us. So after each one of those talks, I will give some minutes for questions, and at the end we can continue doing questions. I will introduce the speakers first, all at once, and then we can go to the first speaker. So my name is
recording in progress.
So my name is Belén Elgoyen, and I'm from Argentina, CONICET. Our first speaker will be Paola Hager from the university, the Catholic University of Chile, the Faculty of Medicine. And she's doctor in neurobiology and research at the neurobiology lab of addiction and behavior. And Paola is part of the FALAN executive committee, and she's going to show us and present the FALAN networking platform. Then we have Marcela Broco from Argentina, from the University of San Martin, and head of the Laboratory of Neurobiology of Stress. And Marcela is going to show us a map that the Argentine Society for Neuroscience has prepared about neuroscience in Argentina. And finally, Cristian Enriquez González Bilaut, who is full professor in the Department of Biology at the School of Science, and also full professor at the Department of Neuroscience at the School of Medicine, and also a professor at the Bach Institute for Research on Aging in California, and the director of El Centro de Gerociencia, Health, Mental Health and Health and Metabolism. And Cristian is going to talk about a Latin American Forum for Research Assessment, also known as DORA. So without any further ado, I give the microphone to Paola. Paola Hager.
Hi, thank you, Belén. Yeah, it's okay. Okay. There are neuroscientists, colleagues. I'm here to present this platform in representation of the executive committee. I am treasurer of this executive committee from 2015, and this is my last year, my last activity. So I'm gonna introduce a little bit our committee. We are composed from Elaine, me, Gloria from Colombia, Belén from Argentina, and Patricia for Uruguay. And this platform was created from Daniel Cava, that he's in change now of the platform and this meeting. And I want to talk about a little bit about our goal to create this platform. We was convinced, like a committee, to create this platform, convinced that we can integrate the community, neuroscience community from Latin America and Caribbean. So now I'm going to present a little bit how this is the function, how we can use this platform through this video.
Welcome to FALAN Networking, the social network of the Latin American and Caribbean Federation of Neurosciences. The platform aims to unite the neuroscience of this part of the world to promote exchange between affiliated societies and promote the development of scientific growth of professionals and students. To participate, you must register by creating a profile by clicking on the Create Account button. Here you must complete your data, study area, location, and your professional situation. Once you confirm your identity by email, you can activate your account. From there you will be able to log in to participate in the groups and forums together with the entire Latin American, Caribbean and world scientific community. Upon entering, you will be able to see the general activity of the platform. You will be able to meet other members, find out what they do, become friends, and exchange private messages. Groups are collaborative spaces for exchange. They are currently defined by countries and by study areas. By entering a group, you can see the wall with information on the members and, in the case of countries, with information on the society corresponding to that country. You can also like the publications that you like and even comment on them and thus generate more interaction. To participate in the groups, you just have to join them. As part of a group, you can enter the forums menu and create your own discussion, debate, or topic. Creating a forum is very simple. You just have to enter a title, a description, and some keywords so that the whole group finds out and begins to collaborate and interact with your proposal. It is also important that you complete your profile with images and information. In this way, other users can see what you do, your participation in the platform, and can interact with you in a more dynamic way. Your profile has an intuitive menu where you can see messages, new friends, complete your profile picture, add links, and upload your papers. Welcome to FALAN Networking. Welcome to FALAN Networking, the social network of the Latin.
Wherever you want. Finally, to improve your experience in the FALAN networking platform, I invite you to complete your profile, participate in the forum, create forum, or participate in other forums created for you. This is the beginning, right? To enrichment your experience depends of you or all of us. Thank you very much. Enjoy the meeting.
Okay, thank you, Paola. I see that there's no questions, but that's probably due that people have to dwell into the system in the networking platform and play with it and see the value of this platform for all Latin America. So I invite all the attendees and those who are not attending, but to spread out the word, and please fill in all the information and go into the platform so that we can start getting all connected. So our next speaker is Marcela Broco with the neuroscience map of Argentina.
Okay, thank you, Belen. Can I share my screen? Is it okay? Can you see the? It's okay?
Yes.
Okay, thank you. Okay, thank you, Belen, for your presentation. I'm here in behalf of the SAN Executive Committee. This initiative started in 2015. There was a survey among the members of the society, and Dr. Guillermo Lanuza, who delivered his talk this morning, prepared and analyzed the data from that survey, and analyze the data and present in this way. This is in our web page. And for the first time, he identified the regional distribution of all the groups doing neuroscience across Argentina, the big areas in which they were working on. And among the 124 groups that responded to the survey, he analyzed and determined the group composition for the society. So this was a very interesting and a very valuable information. So we repeated this year, the survey, and to update this data. And now in 2021, we have more than 200 respondents. And this is our new map. This is our new neuroscience map from Argentina. And from 234 researchers responded. They are more than that, of course. And 226 students and postdocs also answered the question and are included in this map. This map and the following graph were analyzed and performed by Santiago Ojea, a great value and help for us. And these are the main themes that we are working. This is the new composition of the groups with the PhD students, undergrads and postdocs. And this is a new information that we asked in the survey, which collaborations has each group. And these are this graph include national and international collaborations. But all of this information make a picture, a static picture of the map. So we will use this information to create our SAN community. This is a link within our new homepage that will be launched in the next weeks and will be functioning as an intranet for the members of the SAN. And we will find within Comunidad SAN the profile of each member with his or her information, his interests, and also we will be able to upload each of the abstracts that one will be presenting every meeting every year. So all of this information will be a database of keywords that we will be able to use to find members, activity groups, positions, something like a FALAN platform as Paola showed before. And more important, we will use an internal search engine that will be not from Google, from the network of the homepage, and using all of the words that within each profile will be for the search, including the words typed during the debates in the forums. So we believe that some information of the FALAN network platform or the other societies can really help us to create a similar map, but similar to the Argentinian for the Latin American and Caribbean neuroscience. I believe if we update our profiles in FALAN, we can recover the data and make our map. Thank you very much.
Thank you, Marcela. There was a question. People that are not from Argentina some don't know what CABA stands for.
It's the city of Buenos Aires. We have the DC, district. Help me, Belén.
It's the capital of the
country. Yes. Sorry.
Yeah. So very nice work from the society, the Argentine Society for Neuroscience. I think that these maps and also the FALAN network will be very, very useful for all of us. I remember lately that the Latin Academy of Science asked me if I could give them an updated list of all people doing science in Argentina, for example, because they are doing a project trying to oversee what type of science is done in Latin America. And this map from SAN was really, really useful. Of course, I couldn't give them any other information for cancer or whatever, but updating this information both for Argentina, other countries, and FALAN getting all of it together, it's a great leap forward, I think. Okay, yeah. Thank you, Marcela. So now, Christian, it's your time.
Okay, so let me know if you can hear me.
Yeah, we can.
And are you seeing my presentation? So
yes, I think so.
Right. So I'm going to put on full screen mode, and you can let me know whether this is okay. Okay. Well, so thank you very much for the possibility to present what we're doing at DORA. So my name is Cristian González Pilló, and I am part of the DORA Advisory Board, International Advisory Board. So first of all, since I am part of the DORA initiative, so absolutely, I truly advocate that another way for research assessment is possible. And in the next few minutes, I would like to share with you why in DORA we believe we need to move to a new paradigm of research assessment. And first of all, because most of you are really aware that the impact factor is nowadays the best and the most used proxy to assess research quality and research accomplishment. And there's many things about the impact factor that are failed, and we could discuss about that later on. And unfortunately, what it used to be a mechanism or a way of work in science that tell us that you have to publish or perish. Now, nowadays, that transform into impact or perish, because most of the things that we're doing are being associated to the impact factor of the journals that we publish our work. And this is something that we at DORA believe it's a misconception of research assessment. Why is that? So I'm going to give some background about it. So one of the main problems about the journal impact factor, it is that the distribution of citations within journals, it's not evenly distributed. And here you can see, I'm going to put my laser point, give me a minute. Okay, so here you can see the impact factor of the two most important journals on science, which are Nature and Science. Both of them have an impact factor that is close to 40. And you can see here that this 40, which is the average impact factor for the papers published in Nature and Science, it is very much contributed by some papers that have a lot of citations, these peaks which are here toward the right part of the graph. But in contrast, there's a huge amount of papers that are below this threshold. So meaning that here, so of course, the impact factor of the journal, it is not necessarily the impact factor of a paper. And for example, here you have as a comparison +1, where most of the papers are in kind of around this, the average impact factor of the journal, suggesting that here is there is less heterogeneity as compared with this. But the main message here is that it is not the same, the impact of a paper that the impact of a journal, and we all know papers that has been published in Nature and Science that receive very little citations. And this is a problem. The other problem with the impact factor, it is that some of the properties about the impact factor are very specific of a field. So it's not that easy to normalize. So if you compare across disciplines, so the impact factor reflects different trends, and that's sometimes it makes very hard to compare amongst different disciplines. And this second point is very important, that as we humans, because we before being science, we're all human beings. So as long as we are humans, so and any time that you know there is a metric is being used, so there's people that can game or can manipulate that metric. And so basically, so if you put all the interest on quantitative metrics to address or to evaluate scientific productivity, maybe it's not a good idea. And finally, so the calculation of the general impact factor is not really transparent or open to publics, and this is something that also add to some obscurity about the general impact factor. So here I want to mention a couple of results that were published on this paper here on eLife a couple of years ago, because on different universities, the impact factor is being used for many things that include hiring, promotion, and of course, in many countries, it is used for allocate funding by the research agencies. So you can see here 3 types of universities in US. So B types are universities only teach until the bachelor degree. M type are those that have master degrees programs, and R type are more complex universities that have PhD programs. And you can see here that the impact factor is mentioned explicitly on the guidelines to be promoted in those universities. And you can see here that 83% of universities of the more complex use the impact factor as a proxy of quality, and this is particularly relevant to promote people on the scientific career. What is interesting, it is that what the institutions think the general impact factor reflects, and most of them thinks it reflects quality and significant and prestigious. So this is kind of wrong. So DORA, that started in 2012 at the American Society for Neuroscience, and we are addressing the misuse of journal-based metrics in hiring, promotion, and funding decisions. And we have 3 main principles. First one is that we propose that we need to eliminate the use of journal-based metrics in research assessment, or at least if we're not going to eliminate, we need to complement with some other assessment with some other metrics. And we need to foster into a culture that is able to assess the research on its own merit. And we need to capitalize on technology to enhance communication between scholarly and evaluation of impact research. So let me go forward. So how can we do this? And for example, when we talk about science, when we talk about the impact of research, so in addition to the impact factor where our work is published, so we can identify several other areas that could be of interest when we want to evaluate a career, a scientific career. For example, these are we get evaluate how is the contribution of a researcher to develop new protocols. And as a matter of fact, this is a very interesting paper that was published by Van Norden in 2014 in Nature, that revealed that the top 10 papers, the most cited paper on the biology area, that papers that has thousands of citations, the 3 ones that have over 1,000 citations are really technical papers. First one, it's the protein measurement by the Lowry methods. Then it's electrophoresis, SDA page by Lemley. And then it's another way to determine protein concentration in biological sample by Bradford. And if you can see these 3 papers, only one is published on a big journal, which is the second paper that was published in Nature, but the first paper was published in JBC, and the third paper was published in Analytical Biochemistry, a journal that nowadays could be considered not so good. But this paper received over 100,000 citations. So suggesting that it is not the venue where a paper is published, and it's the content what should be considered. So here we have another example, which is about software, and I like very much this example, that is NIH ImageJ. The ImageJ was developed by a computer scientist that was recruited to work at the NIH. And these guys came from an environment that used not PC, but used Macintosh as a computer. And he realized that if we want to develop a platform to analyze microscopy images using a Macintosh computer was not really easy, because most of the technology at that time was related with PC. So basically he came up writing new code for a new software that it's called ImageJ. And ImageJ, after 25 years of analysis, has been used by over 20,000 published papers that use this instrument and this tool. So needless to say that there is a clear contribution of these scientists toward science itself, and this could be not related to a big journal impact paper. So what we do at DORA, so we do some recommendation for funders and research institutes that are here on the slide. So we like that the content of the paper, it's what is most important, not the journal impact factor and of the venue where it is published. And we also suggest that we may consider all the outputs and outcomes generated by research, not only research paper. Of course, we're not saying that research papers are not important. These are very, very important. As a matter of fact, the research paper is the way that we, the scientists, communicate the advance of the knowledge. But we do some other things. We train students, we participate in public policies, we maybe generate a patent that is going to be transformed then into a commercial product. So all of these products should have some way of recognition. So how it is in Chile? So currently 24 of 27 study sections use journal-based metrics at the Chilean agency to provide funding. And however, recently, postdoctoral initiation programs modified their productivity assessment. So this is something that's quite new. So it has 3 years so far. So this is work in progress. And it was going to be very interesting to see whether this new way of addressing or to evaluate research assessment or productivity could have an impact on the way we allocate resources, at least in Chile. So we have some recommendation for publishers, and one of the recommendation is that the publishers should focus on different article level metrics. It's not only how many times the paper was cited, and many of the journals nowadays are moving into that direction, and now you can see that each one of 4 papers, and anytime we access to them. So we can scrutinize how many times this paper has been viewed, how many times has been downloaded, and how many times has been cited. And this is something that it could be sort of interesting, particularly because there is some claim that sometimes scientists are writing paper which no one else is interested in or no one else is reading. And we can say that citation is not the only way to demonstrate that a paper is being downloaded or read by the community. So if we improve and if we enhance the level, the different metrics that we can collect about any specific paper, so maybe we're going to have a more proper panorama of the importance of each one of the papers. We also recommend to publishers that they encourage primary literature citation. And you know, this is something that tends to recognize and to give value to those that have made the findings first. And nowadays it's very easy, and maybe all of us have the temptation to do that, to to support our claims by using reviews on high standard journals rather than citing primary literature. And we believe that recognizing the primary literature is very important to maintain a good culture for assessment for research recognition. And when we talk about metrics providers, so of course we suggest that the metrics provider goes into a new scheme to recognize the metrics that differentiate the primary literature and the reviews. It is very well known that reviews are way more cited than primary literature. So this creates a sort of disbalance when we talk about research assessment. So we believe that the metrics provider may have or should go into a way to discriminate between these two kind of products. And for researchers, of course, we believe that the most important recommendation are to use a range of metrics that show the impact of your work, and that collectively we try to change the culture. So the change is happening. And for example, there's several funders that already started to address and to link to the other recommendation. And here you have an extract from the EMBO postdoctoral fellowships. And now the EMBO postdoctoral fellowship clearly indicate in their guidelines that you don't have to indicate the journal impact factor or any other metrics regarding the productivity of the candidates. And not only that, so in EMBO, so nowadays there is a request that you have to have a paper where you are first author. But if you have a paper when you are first author, you can also put some other papers that are, for example, preprints, which are not published paper, but which they have a DOI and they can be commented by other researchers. So this is a new way just to protect that the scientists can have the primacy on the research. Some publishers are also doing some changes. So here you have an example from development, and here you can see that for a given article you can have different statistics and different parameters or different meters that are more than simply the number of times that the article has been cited. So what we're doing in DORA is to construct some recommendations, and you can see here some of the products that we have developed along these years. So the DORA as a working group started officially in 2018. And here you have two products that you can find at the DORA website. And first one, it's called Rethinking Research Assessment, Ideas for Action. And these address 5 myths that are very common for research assessment, and 5 design principles to overcome those myths. So I'm not going to go into the details of this, but we can talk later about that. And you also describe, and we also are aware about the unintended cognitive and system biases. Anytime we decide to, or anytime that we participate evaluating a paper, evaluating a grant, so all of us are subject to have bias. And having bias, it's not itself bad things, but what we should be aware it is that we have bias, and we have to recognize our bias, and this bias can be of different types, and some of those are presented here on this cartoon. And finally, last June, we launched this rubric that it's called Space for Standards, Process, Accountability, Culture, and Evaluative, that seeks to provide a framework, a very basic framework to analyze institutional progress using some indicators that are kind of standardized. So what is good about this space rubric, it is that it has 3 levels. So you can adjust this rubric for the very beginning to the foundation. So how are you doing things now or in the past, and see whether this way of evaluating adhere to some of the recommendation that we have here in DORA, and then you can move forward into the expansion to increase the movement, and then to scale, because we believe that research assessment, it's a very iterative mechanism that need to be revised sometimes and permanently revised and adjusted. So these recommendations, what is interesting, it is that can be adjusted to funders, can be adjusted to university level, to faculty level, or to department level. It depends on the size of the institution, depends on the size of the group. So the different characteristic that any group can have. And we provide here some background to initiate a discussion that it's, of course, an open discussion that it's going to be expanded in few years more. But we believe that these points here will be instrumental to initiate a movement that seeks to change the culture for research assessment everywhere, in Latin America should not be different. So what can you do? So if some of these things really make you think that this principle could be applicable to your country, to your institution, or to your own group, so I invite you to sign the DORA declaration. So you can ask your institution employer to review the assessment practices. And there's a lot of group practices and examples that are in the web page of DORA that probably will help to implement some of the changes that we are suggesting. And of course, we will be eager to hear about your own experiences. So we all of the products that we are developing at DORA are made for the people to use, for the colleagues to use, and we will be more than happy to hear the outcomes or the conclusion or whatever you may want to comment about the principles that DORA is pushing to enhance research assessment. So this is, sorry for the typo, this is September 2021. So DORA declaration has been signed for over 2,000 organizations and for over 18,000 individuals among the organizations. So we are very proud that we have two major institutions that are supporting the DORA claims, which are EMBO and the Howard Hughes Medical Institute. So we believe that they nicely recognize the value of trying to discuss what are going to be the new assessment for research and what can we do basically, and I'm finishing now, to be very sure that when we are evaluating colleagues and young people and researchers, either for hiring, for promotion, or for funding allocation, we are doing on a very transparent and fair manner. So with that, I will stop here and thank you so much for your attention.
Thank you, Christian, for that very wonderful talk. It makes us all think. And I have a few comments from people in the audience and questions. First, I have Flavio Solesi. He's congratulates you on your talk, Christian. And he says that FALAN meeting, during FALAN, we are hopefully launching a set of a code of conduct involving behavior of participants in FALAN related meetings. So Flavio thinks that research assessment and a full embracement of DORA principles should be the next step in promoting equity in neuroscience research in our region. So it's just a comment and something for the future executive committee at FALAN to take note and work on it as to embrace DORA within the region. And then Pablo López has a question for you, Christian. Do you think that in our countries in Latin America, we don't have a critical mass for evaluating scientific merit of research, and that's why we, or in general, it's tempting or it is easier to go to journal metrics?
Yeah, I think this is true. So one of the things about the quantitative assessment, it is that it's fairly easy. So you need to count points and you can include some adjustment, whether the author is corresponding author or not, first author or not. So you can you can make some connection that make a very easy equation or polynomial to make to a number that allow you to discriminate between different researchers. But what we're claiming on at DORA, it is that we can use journal impact factor at some point, but it hasn't to be the one and the most essential criteria. So our suggestion is that when we, particularly for recruiting, that we do what the people used to do many years ago. So you have to read the papers of the applicant. You have to read the papers of the applicant. You have to identify what are the strengths of those papers. You have to ask the applicants, what is their contribution, or how do they get into those ideas? What were the problems that they need to face and to solve to get into their conclusion? So it's more an holistic approach. It's not only the venue and the number of the paper of the impact factor. So the trend nowadays is moving in many places, particularly in Europe, towards an evaluation on the productivity that is small, that is more related with what it is called the narrative resume, where the applicant has to describe himself or herself why and what is the importance of their contribution to any single field, rather than simply mention the papers. And I think this is a good way to track changes and to track forces into a new way of assessment. So regarding the number of people that can be involved into evaluation, I agree with Pablo that we are a small community in all of our countries, that very few people tends to be linked to all of aspects of research, going from assessment to funding allocation to conducting research. And this is something that we need to improve. Of course, we need to open space to more people. So we need to open space that all of us, all active researchers, can have the possibility to get involved into research assessment, for example, as evaluator of grant proposals. But that, I think, needs that all the people should at least receive a little bit of formal training on how to evaluate a grant. Sometimes, and this is very common here in Latin America, when you are recruited, when you are hired on a university or a research center, so basically you get lab space and that's it, and you have to start using your intuition and what is going on to research, to teach and to evaluate some other colleagues. And maybe this is a good time to discuss whether all the active researchers should receive some training that allows them to at least have a common perspective of how research should be evaluated. And in that way, so probably this critical mass that Pablo is referring could be improved.
Let's see if there's another question. If not, I have a question. No. So I'm in several committees and at different standards, and some are prices, and there in prices, it's more important to really identify what's the contribution of the person to the field, and that's easier. And then you can have letters of references of people who really identify what they have done. But at lower levels, I mean, when you're recruiting or when you're assessing, what we have here is to come into CONICET, we have so many applicants that you need some sort of standardization. It's not only impact factor. What we do here is we scale the impact factor to the area of research. So for example, by SIMAGO. So that it's not the same impact factor, for example, in ecology or cancer. Cancer has higher impact factors, but we take into account several things. Where do you publish? What have you published? Not the impact factor of the journal, but the relative impact factor. How many citations? I think that in general, how many times a paper is cited within the field is also important. It's not the same being cited in ecology that probably in cancer. You can't compare them, but it's a difficult issue, no, Christian?
Yeah, I totally concur. Of course, this is not a simple task, and of course, it requires that many people should be involved. And of course, whatever the metric you choose, there will be some drawback about it. So whether it's impact factor, the H index, the citations. So we could discuss about a lot of things that could be wrong if we simply focus on these three, which are very common nowadays in the research panorama. But what I think it is worth mentioning is that I would say that one of the things that it is very helpful is try to discern very well, very appropriate. I'm not exactly sure how this could be done. What has been the evolution of each one of the candidates, rather than to compare side by side, just to compare to themselves 5 or 10 years ago, how they have evolved. Because I think this is a very good predictor of what is going on, what is going to happen later on. So, and in a sense, it's more fair. So when you are competing against yourself, so there is no bias because you are competing to yourself. And maybe that it's a good strategy. I'm not saying this is the solution to this conundrum, but I think this provide another level of assessment that could be perfectly complementary to those metrics that we're using nowadays.
Yeah, what is your slope? Are you climbing or are you going down?
Yeah, absolutely.
No, that's true. Yeah. Let's see if there's another question. If not, there was a question. Yeah, Juan Ferrario, great talk. Do you think, well, you mentioned already, but do you think, Christian, that H index is more reliable or also has a bias?
So there is one single thing about the H index, and it is that the H index grows as you get older. So as long as you get older, your H index is going to be growing. So there is some ways to overcome this, and there is one specific way that it is called the M quotient. And the M quotient, it is exactly the H index divided by the number of years where you published your first paper. So basically, it's just you're calculating the slope of your productivity, you know? And in that way, so of course, if you do that, so you can, sort to equalize the age distribution, because basically all of the researchers are going to be compared to the very same. So because it's going to be the slope of their productivity. This way, I think it's better that age index, but it's not everything at all, So my message to take home, my take home message today is that there shouldn't be one single concept, one single quantitative measurement that indicates the quality of our researchers. So I think we need to, of course, it's more demanding, it's more challenging, but we think we need to spend a little bit more time, as it was in the past, because in the past it was like that. So there was committees that study quite a bit about all of the candidates and spend a lot of time doing that. And I believe we should try to combine both quantitative and qualitative assessment to reach into a final conclusion that in the very end convinces all of us that we're doing a fair job.
Yeah, I agree completely. H index alone also, I mean, you can have lots of collaborations and have a huge H index and never be a first or last author. So it has to be in conjunction with other. So if there's no more questions for Christian, there's comments in general. Pablo López says that it's critical that in the FALAN network we have search engines, tools for keywords, themes, etc., that allows quick search of topics. And I think that has been included, Pablo, but we can check with Daniel. Another question, Paola, is how do we encourage people to update the profile? And I think that, I mean, the FALAN network is sort of recent. We just started it, launched it very recently, but we have to do an active way of promoting and people getting to know the network and encouraging them to put their profile. in there. So that's work that we need to think of. And no, Paola, what do you think?
Yeah, it's not hard to put. If you are inside, already we have 800 people inside of the platform, and I think 10% have the full profile now. So yeah, if we start to put our papers, our information in the profile, the connection is going to be easier, and it's going to be expanded to outside of the network to include other people. I think it's work that we have to do in general, the community, right?
Yeah, it's a little bit of work, but we have to do it. It's important. Like me, I think that in my profile, there's only my name. So I'm the first one to go in there and update my profile. Okay, I think that we're almost on time. So I want to thank Paola, Marcela, and Christian. I think it has been very encouraging, 3 different topics. And we didn't not only get to know the FALAN networking and getting to know FALAN, but we get to know about this very interesting DORA project that probably leaves us thinking a lot and trying to analyze how we do evaluations, and also very nice, Marcela, what the Society for Neuroscience is doing, and seeing how the map is progressing from the first map that Guillermo Lanuza started, and now we have all this new information in there. So I want to thank the 3 of you and invite people to continue, stay tuned, because this afternoon session continues. Thank you very much.
Welcome everybody to this plenary lecture. I am Cristian González Pillo. I am the president of the Chilean Society for Neuroscience. And it is my great honor and pleasure to introduce today Professor Cecilia Hidalgo, which is not only an outstanding scientist, but also a very, very, very good friend. So Cecilia was trained as a biochemist at the University of Chile back in the 60s. And then she has the merit that was the first female researcher to get the PhD degree at the University of Chile close to the 70s. After that, she decided to go to US after receiving a Fogarty Fellowship, and she was working on the NIH in the US for 4 years. And then she went back to Chile until 1974, when she decided to go to US again. And she was based this time on the Boston area. In first term, she was working as a staff scientist at Boston Biomedical Research Institute in Massachusetts. And then she had an appointment as associate researcher at Harvard Medical School. In the middle 80s, she decided to go back to Chile, and she was hired, she was recruited at the Faculty of Medicine of the University of Chile, where she became full professor in 1986. In 1993, she received a Guggenheim Fellowship, and then between 2001 and 2003, she served as the director of the Cellular Molecular Biology program of the Institute of Biomedical Sciences at the Faculty of Medicine, University of Chile. She is currently part of the Department of Neuroscience and of the Physiology and Biophysics program at the Institute of Biomedical Science at the Faculty of Medicine. So she has made a lot of work in addition to research that it involves a lot of commitment to society activities. And she was actually the president of the Chilean Society of Biology, and she was also the president of Society of Latin American Biophysicists, and she was also council member of the Biophysical Society, the International Union for Biochemistry and Molecular Biology, and the International Union of Physiological Sciences. She is, of course, member of the Chilean Academy of Science, and currently she's the president of the Chilean Academy of Science. So this is a great recognition to his work in our country. So in 2004, she received a very selective award at University of Chile, that is the Amanda Labarca Medal of Merit, that is given to female academics that has made outstanding contribution to the country. And in 2006, she received, and she was the first woman to receive the National Prize on Natural Science in our country. Interestingly, until the late 90s and early on the beginnings of 21st century, so she worked mostly associated to calcium signaling in muscle cells, and then she moved and she shifted her interest toward neuroscience, and currently she's studying communication between neurons, and she addressed the role of calcium as a signaling molecule and how calcium is connected with redox biology, and how these two seemingly unconnected events can regulate neuroplasticity and spatial memory. She has published a lot of papers, over 100 papers. She has received a lot of citations, over 8,000 citations. And she very recently published a very interesting paper on PNAS that described mechanisms for calcium signaling propagation to the nuclei in hippocampal neurons. So on a more personal note, Cecilia is married to Marco Tulio Núñez, and she has 4 children and has 5 grandchildren. So it is a pleasure for me to welcome you, Cecilia, and now I give a stage to you.
Recording in progress.
Well, thank you very much, Christian, for that very kind introduction. And thanks very much also to FALAN Networking 2021 for inviting me to give this plenary lecture. It is really a privilege for me to share our recent work with you, especially because I am working on a field that is kind of a secondly rated field in neuroscience. And since 1999, and here I have a graph going from right to left, showing the number of papers that have been published. This is a PubMed search on ryanodine receptors and neuronal function, and neurons really. And you can see that many, many papers have been published along these years, yet the role of ryanodine receptor-mediated calcium release on neuronal function is not widely acknowledged. If you see reviews, current reviews, you see that there's a neurotransmitter release at the synapses, and that generates calcium entry into the postsynaptic terminal that then hits local targets and calcium-dependent signaling pathways that then stimulate neuronal plasticity and memory processes. But the role of intracellular calcium channels, such as ryanodine receptors, on this signaling is not generally acknowledged, even up to date. And as an anecdote, I'm just reviewing a paper in which it says that ryanodine receptor function on neuronal function is still not known. And the topics of this talk, I hope, will kind of show you that that's not quite the case. So what I will address today is how ryanodine receptor-mediated calcium-induced calcium release, CICR, in hippocampal neurons is first redox sensitive. It's also required for synaptic plasticity, LTP, LTD, and dendritic spine remodeling. It's required for spatial memory processes, amplifies calcium transients mediated by IP3 receptors, and plays a major role in the generation of activity-induced nuclear calcium signals. As you all know, calcium-induced calcium release mediated by ryanodine receptors is absolutely essential for cardiac muscle function. You have a small calcium entry mediated by voltage-gated calcium channels at the plasma membrane in cardiomyocytes that generates a local calcium increase that is sensed by neighboring ryanodine receptors that then are opened by calcium and generates a calcium increase in the entire cytoplasm that allows a muscle construction. And that was described, this calcium-induced calcium release process, by Fabiato and Fabiato in Nature in 1979. And I would like to go back to 1998, when Sir Michael Brevich published in Neuron a very important article titled Neuronal Calcium Signaling. And in that article, and here is Sir Michael Brevich, and there we are together at a meeting in Europe, and he explored the idea that the endoplasmic reticulum and the plasma membrane form a binary membrane system that functions to regulate a variety of neuronal processes, including excitability, associativity, neurotransmitter release, synaptic plasticity, and gene transcription. And here is a scheme taken from his paper, his review paper, showing that DER is kind of a neuron within a neuron, the neuron within a neuron concept that he coined. He also proposed, this was a really visionary work. He also proposed that calcium entry generates, through different calcium channels at the plasma membrane, generates calcium signals that activate IP3 receptors and ryanodine receptors, and that there is crosstalk between these two kinds of calcium release channels via calcium-induced calcium release. Even more, he proposed that calcium signals generated either at the spine, at the dendritic spine, or at the soma, are propagated towards the nucleus, towards the neuronal nucleus, via calcium-induced calcium release as a calcium wave, a regenerative calcium wave, as you see here, either from the soma or from the faraway dendritic spines. And not only that, he also proposed that these calcium signals propagate to the nucleus, where they serve to activate the CREF-mediated gene transcription. So we have explored, and I will show you in this presentation, several of these proposals. First of all, I'd like to show you that ryanodine receptor-mediated calcium-induced calcium release in neuronal cells is affected by the neuronal redox state. It's redox sensitive. And the story started in the late 90s, when Juan José Marengo and Ricardo Bull and myself, we did some experiments in which we incorporated ryanodine receptors in a lipid bilayer, where we registered currents, calcium currents, through the channel, and we found that if we incorporated single ryanodine receptor channel from rat neuronal cortex, we found that we had 3 different responses to changes in the cis calcium that corresponds to the cytosolic calcium site of the channel, the cytoplasmic calcium site. We had some channels that barely responded to calcium. We called them the low P0 channels. And then we had channels that responded to calcium with a bell-shaped response, activation by calcium and then inhibition. And then some channels responded to calcium by with a activation and lower calcium concentrations, and then if we increase calcium to the 700 micromolar level, we still didn't have inhibition of the channel. And it was tempting to speculate that neurons from the rat cortex had the 3 ryanodine receptor isoforms, so that this would correspond to the response of the different isoforms present there. Because we know that mammalian cells have RYR1, RYR2, and RYR3, type 1, type 2, type 3 ryanodine receptor channel isoforms. But we found that what was originating these 3 different responses to calcium, and it was absolutely clear that there were 3 responses. Either the channel responded with low activity, with bell shape, or with high activity, and highly reduced channels responded very poorly to calcium, partial SH oxidation of cysteines, and then I'm not going to go into the details of that, but partial oxidation generated this bell-shaped response, and then further oxidation of the channel here generated this high open probability response that the channel was activated by low calcium concentrations and was not inhibited by increasing calcium up to the 700 micromolar range. So the question was, is this happening also in neuronal cells? Because one thing is to show that the single channel is affected by the redox state, but then another very different thing is to show that this also happens in neuronal cells. So how we tested the role of ryanodine receptor-mediated calcium release of neuronal function? We use primary hippocampal cultures from male rats, and we suppress ryanodine receptor activity with inhibitory ryanodine. Ryanodine is a plant alkaloid that is present in an Amazonian plant, Rhyana speciosa, and it has an interesting way of acting with the channel. If the concentration of ryanodine is less or equal to 1 micromolar, it binds with high affinity to the ryanodine receptor channels and activates channel opening. However, at concentration higher than 10 micromolar, it binds preferentially to the open channels and suppresses their activity. Because the channel needs to be open in order to bind high concentration of ryanodine, to get complete inhibition, it has to be incubated for long periods, 1 hour or more. So you need high concentration of ryanodine and long incubation periods in order to get complete inhibition of ryanodine receptor activity. And I will quickly show you this, that we have a record in which we're checking calcium signals in hippocampal neuronal cells. And we see that when we add ryanodine 50 micromolar, initially there's no signal. And then after 1 hour, if we add an agonist, specific agonist of ryanodine receptors, we don't have a calcium signal in response to that. And that's already shown here, in which the control cells respond to the agonist with an increase in calcium, but the neuronal cultures, the cells from the cultures that have been incubated with inhibitory ryanodine did not respond to the ryanodine receptor agonist. And furthermore, the question was, is it because the ER was depleted of calcium by incubation with inhibitory ryanodine? And here we show that that's not the case, because if we add tapsigargin that inhibits the SERCA to the cultures, we find that the neuronal cells respond to tapsigargin even when they have been incubated with ryanodine for a long period of time with inhibitory ryanodine. So treating the neuronal cultures provokes complete inhibition of ryanodine receptor-mediated calcium release in neuronal cells. Then we went on to show that ryanodine receptor-mediated, say, CICR in primary hippocampal neuronal cells is redox dependent. And here I'm showing all the people who did this work. Tatiana Alasme, Maria Angelica Carrasco, Ulrike Kemmerling, Denise Riquelme, Alexis Sumedes, Andrea Paula Lima, Gina Sanchez, my husband, Julio Núñez, and my brother, Jorge Hidalgo, very much in the family, as you can see. And we found that when we added hydrogen peroxide to culture neuronal cells, we elicited calcium-mediated, ryanodine receptor-mediated calcium transients that enhance the S-glutathione of the protein. And you can see here that these are neuroblastomal cells, and these are hippocampal neurons, and we added hydrogen peroxide. These are line scans. We have in the control cells an important increase in calcium, cytoplasmic calcium. But if the culture had been incubated with inhibitory ryanodine, this response did not occur. So we know now that a redox agent like hydrogen peroxide elicits ryanodine receptor-mediated calcium release and also causes reversible cysteine modifications in the channel protein. And then we show also that ryanodine receptor-mediated calcium-induced calcium release stimulates calcium-dependent signaling pathway. And here I have some of the people who contributed to this work that I mentioned already. And when we added hydrogen peroxide, we found that we had an increase in ERK phosphorylation and in CREB phosphorylation. And this happened also not only in primary hippocampal cultures, but in hippocampal slices as well, in which we have an increase in phosphorylation of CREB and the phosphorylation of ERK. And moreover, additional hydrogen peroxide leads to ryanodine-mediated activation of the transcription factor NF-κB, as you can see, NF-kappa B. As you can see here, when you add hydrogen peroxide, you have a stimulation of a reported gene for NF-kappa B that is completely inhibited with ryanodine, inhibitory ryanodine. Then we went on to stimulate with high-frequency field stimulation the cultures, and we found that that elicited redox-sensitive ryanodine receptor-mediated calcium-induced calcium release. Here we have in the control the black line in which high-frequency field stimulation, 1,000 pulses at 50 Hertz, induced first a small signal, calcium signal, cytoplasmic calcium signal, followed by a large increase in cytoplasmic calcium that was completely abolished by pre-incubation with inhibitory ryanodine, and was also inhibited by pre-incubation with the antioxidant agent N-acetylcysteine. So we are showing here that this is due, this amplification of calcium entry through NMDA receptors. I'm not showing that, but this is due to NMDA receptors. This first calcium influx, calcium increase in the cytoplasm, that it requires functional ryanodine receptors, and that it doesn't happen if you have increased the reduction of the neuronal cells by addition of this reducing agent N-acetylcysteine. We also found that high-frequency field stimulation of primary hippocampal culture induced cross generation. And you can see here a record of the detection after applying 1,000 pulses, 50 Hertz, and we detected cross generation with a hypercytoprobe. And you can see how the fluorescence of the probe increases. Here is a record. This is one experiment. This is the type of culture we use. And we found that just cross generation by high-frequency field stimulation, by activation of neuronal activity, depends on calcium, is abolished when you add BAPTA-AM, depends on the NADPH oxidase NOX2, and requires calcium entry via NMDA receptor because it's inhibited by MK801, and it's also inhibited by ryanodine. So here we know that it's calcium-induced calcium release that provokes this cross generation and an activation of the NOX2 NADPH oxidase isoform. It also, high-frequency field stimulation, stimulates the calcium-dependent translocation to the nucleus of the P65 subunit of the NF-kappa B complex. And you see here, the control doesn't have any fluorescence in the nucleus. When you stimulate, you have a translocation to the nucleus of the green fluorescence. But if it is inhibited with ryanodine at micromolar concentrations, this translocation to the nucleus of the P65 subunit does not occur. So if we look at the schematic description of the pathways we think are operating here, we think that high-frequency stimulation stimulated NMDA receptors, there's calcium influx, activates calcium-induced calcium release by ryanodine receptors that generates calcium signals that stimulate the NOX2 isoform, and calcium also induces the neuronal NOS isoform that we know has implications also for activation of the NADPH oxidase. And this crosstalk between calcium and ROS induces several responses in neuronal cells. It promotes ERK and CREB phosphorylation and activates the transcription factor NF-kappa B. Then we went on to to investigate whether a Ryanon receptor is required for synaptic plasticity, LTP, LTD, and dendritic spine remodeling. And we did this work with Tatiana Dafne, Alejandra Arias-Caviedes, Pablo Muñoz, Genaro Barrientos, and Jamila Moore. And I don't have to tell this audience what synaptic plasticity, LTP, and LTD are. And dendritic spine remodeling, also you know very well these responses. If we induce hippocampal LTD with theta-burst stimulation for trains, here is the response of the control, a very robust LTP. But if we pre-incubate with inhibitor of ryanodine, this is very much suppressed, this response. And here is another protocol, another experiment for a shorter time record. This is only 1 hour, these are 3 hours. And we found that when we inhibited ryanodine receptor-mediated calcium release with ryanodine, we did not affect the pair pulse response, suggesting that this inhibition was at the postsynaptic level. We also found that activation of ryanodine receptor, remember that I told you that low concentration of ryanodine stimulate ryanodine receptors, then we added 1 micromolar ryanodine, and here we have the activation of the LTD response with 1 micromolar ryanodine, but when we incubated with 10 to 20 micromolar ryanodine, we didn't have any stimulation of the LTD response by low frequency stimulation. And we found the same thing with caffeine, that when we added caffeine, we also stimulated LTD. That caffeine is also an agonist, less selective than 4-C of the ryanodine receptors. And then we went on to look at dendritic spine remodeling, and here is a sequence of images taken at different times in which we added BDNF, the neurotrophic BDNF, to the primary hippocampal cultures. Here we have a prolongation of the neuronal cell, and you can see that 30 minutes, 45 minutes, and 60 minutes after BDNF addition, we have the generation of a new spine. However, the cultures treated with ryanodine did not respond at all, and these are short-term time courses. And if we look at longer-term records, 6 hours with BDNF, we have the control here, dendritic prolongation, and here the response to BDNF plotted there for many cultures. And when we added ryanodine, we pretty much eliminated this response. So somehow in the signal transduction pathways initiated by BDNF, in some part, ryanodine receptor-mediated calcium release is very important for that. We know, and it had been published before, but here are our own determinations, that ryanodine receptor type 2 is the main isoform present in both in hippocampal tissue and in the whole hippocampus. If you compare it in the hippocampal culture with the hippocampal tissue, you know that ryanodine receptor type 2 is the isoform present. This is at the protein level, and the type 3 isoform is present in much lower proportion, and the IP3 receptor type 3 is also present at lower levels. And importantly, we have not been able to find the type 1 isoform in our samples of primary cultures, or even in the intact hippocampus, in the homogeneous of a whole hippocampus. And the messenger RNA is present at such low levels that is within the signal-to-noise ratio. And then, since RYR2 is the main isoform, we transfected neurons with an SH against ryanodine receptor type 2, and we found that it has, both in the soma and in the dendrite, a significantly decreased expression of ryanodine receptor type 2, and also in the whole culture in which we have transfected and non-transfected neurons, we found that the expression of the ryanodine receptor type 2 at the protein level is also decreased compared to the control. And then we investigated what happened in these neurons that have been transfected with the SAGE-RYR2. And we found that they did not respond. We know that they have been transfected because we had also a red fluorescent probe. And we found that these neurons did not respond to the agonist CMC. They also did not respond to when we inhibited the generation of nitric oxide with LNM, and did not respond when inhibited the NOx2 isoform. So we are showing here that transfecting neurons with the SAGE-RYR2 inhibits the agonist response, and also that inhibition of the generation of nitric oxide or superoxide anion by NADPH oxidase prevents agonist-induced calcium release. So again, we are showing here that we have mostly calcium-induced calcium release by ryanodine receptor type 2 isoform, and there is redox sensitive. And also transfection with this SAGE-RYR2 that decreases the ryanodine receptor expression in the transfected neurons, inhibited the response to BDNF and dendritic spine remodeling, induced either by BDNF or by caffeine that also induces spine remodeling that is inhibited by the transfection with the SAGE-RYR2. And also N-acetylcysteine inhibits the response induced both by BDNF and by caffeine. Again, showing that if we want to have successful ryanodine receptor-mediated calcium release, we need a certain redox tone in the neuronal cells. Here are some longer time records showing essentially the same response, that when we have transfected with the scrambled sequence the neuronal cells, we have a robust response to the neurotrophic BDNF in terms of the dendritic spine remodeling, and that it doesn't happen in the neurons transfected with the SAGE-RYR2. And the same in the response to caffeine. Then we went on to study whether spatial memory processes require ryanodine receptor-mediated calcium-induced calcium release. And we did these experiments with Paola Heger, with Alejandra Alvarez, Yami Lenore, and Jose Luis Valdez. We injected intrahippocampal stimulatory ryanodine concentration that when we did the test, we knew that because we injected also radioactive ryanodine, that it had reached the 1 micromolar range. And we found that 24 hours, when testing the animals, 24 hours after injection and for several days in several sessions, we found that the animals injected intrahippocampus with stimulatory ryanodine had a lower escape latency along the sessions. But this effect was transient because 96 hours after having injected the animals, this stimulation had disappeared. And also the animals injected with the stimulatory ryanodine had a more consolidation of the memory than the control animals. And we also evaluated hippocampal-dependent spatial memory in the OASIS maze task, which involves searching for a reward, in this case it was water, present in 1 of 21 equidistant wells in a circular arena provided with visual clues. And the memory task is finding the reward after training, and the well position was constant in all sessions. So we trained the rats for 3 days in the OASIS maze task, and then at day 4, we injected the first injection of ryanodine, and we did another session. We injected we injected an oligodeoxynucleotide against RYR2 or the scrambled sequence into the CA1 region. And we did 3 injections as described here, and a total of 9 sessions in 7 days. And after the 9th session, we removed the hippocampus and checked what was the RYR2 content in the hippocampal region. If we test the rats at session 9, we find that rats injected with the scrambled sequence, with the ODN scramble, you see that they did very well in finding they were placed here, for instance, in the trial 1, and found the reward very easily at different trials. However, the rats injected with ODN RYR2, they had a hard time finding their reward in all trials. And sometimes they just didn't find it. And here we have a summary of the number of errors of the 2 different groups injected with RYR2, the scramble sequence or the ODN. And we found that after the injections, the number of errors increased in the injected group with the ODN RYR2. And this is a graph here, the average values, and also the injected rat display an increased ratio of the distance covered to reach the reward compared to the control group. So these results indicate to us that injection of the RYR2 antisense oligonucleotide in the hippocampal CA1 region impairs spatial memory. And after the 9th session, we sacrifice the rats and we check the RYR2 content. And you see this is a immunofluorescence, and this is the CA1 region of the rats, from the rats injected with the scramble sequence. And you see that the ones that were injected with the ODN RYR2 had a very much decreased content of the RYR2 isoform, but not of the RYR3 that remain constant in all 3 groups. So we know that with this maneuver, we're really decreasing specifically, selectively, the RYR2 isoform. More recent work, I'm going to show you, again, going back to the review by Sir Michael Burridge in 1998, in which he proposed, as I described to you, that IP3 receptors release calcium that can activate ryanodine receptors. And here is work done in collaboration with Stephen Hartel, Andres Cook, Mauricio Serra, Pedro López, Alex Córdoba, and Omar Ramirez, who's the first author of this work. And here's to show you that hippocampal neurons culture express the RYR2 isoform and high density, and also the IP3 type 1 isoform, the IP3 receptor type 1 isoform. And here is the colocalization of the 3 labels we have. We have this red as an ER marker. And we found that if we induce the release of IP3 by uncaging IP3 present in the neuron with UV pulses of UV light, we generate a calcium signal, and I hope this runs, in which you can see that there's a calcium signal that is propagating along the dendrite. And here is a time course and the propagation of the signal. And if we inhibit ryanodine receptors in this culture, and then we engage IP3, we found that the latency of this signal emergency is not altered by the inhibition of ryanodine receptor, but the time to peak is increased, and the maximum amplitude of the signal is decreased. So this shows that these 2 receptors, these 2 calcium, intracellular calcium channels talk to each other, that calcium transits evoked by IP3 receptor stimulation is sensed by ryanodine receptors and contributes to the signal. It contributes to increasing the appearance of the signal, and also it contributes to its maximal amplitude. And I'm coming near the end of this talk, I'd like to show you that neuronal stimulation generates calcium signals mediated by remodeling receptors that reach the nucleus. Again, coming back to the review with Sir Michael Lourenco in 1998, in which, as I showed you, proposed that the calcium signal generated at the dendritic spines or the soma reach the nucleus. And this work was done by Pablo López, Alex Córdoba, Ignacio Vega, Tatiana Dahme, Omar Ramírez, Jorge Toledo, Mauricio Cerda, Stephen Hartel, and Andrea Paula Lima. How do we stimulate primary neurons in primary culture? We use three different strategies. We have the GABASIN, which is an inhibitor of GABA-A receptor that stimulates degeneration. This has been published in the literature before, primarily by the group of Hilmar Bading. And this induces calcium signal in the cytoplasm that propagate to the nucleus. We also use glutamate anti-aging with UV light into a dendrite. And we also use, which I'm showing you already, the high-frequency field stimulation protocol. And we detected calcium signals with fluorophore or the neurons that were transfected with a cytoplasmic calcium sensor, GCAM-P5. And we detected nuclear calcium signals by transfecting neurons with a nuclear calcium sensor, GCAM-P3 NLS. First, I'm going to show you responses to GABASIN. And GABASIN addition generates calcium signals in the cytoplasm and the nucleus. And of course, I cannot write the movies much as I tried before. Well, let me see if I can do this. Maybe if I take off. I don't know why, because I checked them before and I could run the movies, and I really would like to show them to you how the calcium signals are really synchronic.
Cecilia, maybe if you disable the pointer, you can
I'm trying
activate
to
the
do
videos.
that.
Yeah.
I'm trying to do that, but I cannot
It
do
is,
that.
no, one more to the left.
Okay, let me see.
It wasn't that.
That one?
One more to the right. No, to right. There. There.
I cannot disable it.
Okay, so.
See, I'm trying to do it, but I cannot do it.
All right.
I'm really sorry about this.
So here it's to just.
So how can I disable the pointer?
You can use the escape key on your keyboard.
Yeah.
And now you should be able to run videos.
Yes, thank you. So there you are.
Lovely.
Thank you, Kristen. There you have the effect of GABASIN and cytoplasmic calcium signals and on nuclear calcium signals as shown here. This is the sensor, the calcium sensor in the nucleus, and you can see that in a culture at low amplification, all the neurons are responding synchronously. And this is seen here. This is a record from several neurons in the same culture. After following additional GABASIN, this is the average record, the lower part. And I'm not going to show the pointer again. And if you compare to the right, when you add gabazine to a culture pre-treated with ryanodine, the neurons do not respond or respond very poorly. And all these synchronous signals elicited at later times are fairly much gone. And here I'm showing you the effect of ryanodine on that, and how it decreases the maximal intensity of the signal and the area under the curve. And going back to what I showed you before, transfection with the SH-RYR2 also decreases very much the response to gabazine compared to the response elicited by neurons transfected with the scrambled sequence. And this is a graph in the lower part of this image, in which you see how the intensity of the signal is very much decreased by transfection with SH-RYR2, and so is the area under the curve. I will show you now what happens when we do local glutamate engaging in the dendrite, and you can see that by doing 5 pulses of engaging, this is the nucleus in the top part that has the nuclear calcium sensor, and you can see that 10 pulses already originates a signal at the nucleus, and spontaneous activity generates important increase in calcium in the nucleus, and then 20 pulses generate another increase, a high increase in the nucleus. And I would like to show you here what happens when we do this stimulation of glutamate engaging. And you saw in the movie there, first an increase, then a higher stimulus, a further increase in the nuclear calcium signal. And if we compare the response with neurons transfected with the SH-RYR2, you see that the responses are very much decreased to the different stimulus that cause a significant response in the control neurons. And this is plotted there at the right. And then if we do high-frequency field stimulation, we find that we have an increase in the nuclear calcium signal when we do a higher stimulation with 100, 200, 300 pulses. And you can see here that in the movie at lower left, that if you look at the control, 1 stimulation and 2 stimulation, very robust calcium signal. When we do it the same with ryanodine, you have a very low response. And this is plotted here with different stimulation protocols. In all cases, the neurons in cultures treated with inhibitory ryanodine have a much lower nuclear calcium signal than the control neurons. And the same happened with the culture with the neurons transfected with the SH-RYR2, in which you can see the response of the control to different stimulation, high-frequency field stimulation protocols, whereas the one transfected with the SH-RYR2 had very reduced responses. And then, and this is the last topic I'd like to talk to you today, to show you today, is that Berich again in 1998, in this very insightful review, he said that the nuclear calcium signals originated via calcium-induced calcium release would stimulate KREP-mediated gene transcription, would stimulate KREP phosphorylation and KREP-mediated gene transcription. And we found that inhibition of ryanodine receptors with ryanodine decreased the enhanced KREP phosphorylation induced by gabazine. You see here, and it also, when you look at the graph on the right, it also decreased very much the basal level of phospho-KREP when you stimulate when we treat the cultures with inhibitory ryanodine. So you see here that ryanodine, inhibition of ryanodine receptors somehow decreases the stimulatory effect of gabazine on the phosphorylation of the transcription factor, of the nuclear resident transcription factor KREP, and also decreases the expression of MPAS4, which is a neuronal activity-dependent immediate early gene that has been identified as a transcription factor that is very relevant for memory formation. And it's known that this expression requires nuclear calcium signals, and you see how neurons in calcium treated with inhibitory ryanodine have a decrease in stimulation by gabapentine of the MPAS4 mRNA levels. And surprisingly, also gabapentine stimulates the mRNA levels of RYR2 isoform, and is also inhibited by ryanodine itself. So there's a loop here that calcium released by ryanodine receptor seems to stimulate also its own transcription. So in conclusion, I think we have demonstrated that redox-sensitive ryanodine receptor-mediated calcium-induced calcium release in hippocampal neuronal cells stimulates calcium-dependent signaling pathways that are important for synaptic plasticity and memory, plays a significant role in hippocampal synaptic plasticity and spatial memory processes, and that it contributes to the propagation to the nucleus of activity-generated calcium signals that promote gene expression. And you could see that it's not absolute inhibition. So there are other ways in which calcium signals reach the nucleus, but ryanodine receptor-mediated calcium release has a significant role in this process. And here I have already mentioned all the people that have collaborated along these years in developing this work. And we have had two international collaborators, Dr. Phil Mulvaney in Germany and Dr. Eric Plant in the United States. And I'd like to thank the technical help of Dr. Luis Montesino, J.P. Filkenstein, Jose Galaz, and Nicole Enriquez. And we have had support from FONDACIT, Regular and FONDAP, Millennium Science Initiative to the DNI, and we have an international cooperation project, Chile, Germany. And thank you very much for your attention.
Thank you very much, Cecilia, for your wonderful talk. So we have time now for questions. So we remind the audience that you can leave your questions on the chat zone. And I'm going to go to revise some of the questions which are already there. So the first one came from Paula Lunardi, and she's talking, she's asking, is it possible that activation of ryanodine receptor is involved in memory decay or forgetting mechanism?
Yes, I think it's quite possible. We have to go into that because if we know that ryanodine receptors are important for LTD, so somehow I think they are also involved in forgetting memories, that we have to study that. There are so many things we have to study. What are the intermediate pathways that are important, for which ryanodine receptor-mediated calcium signals are so important for these cognitive processes and for synaptic plasticity that we know is also important for memory.
The next question is from Osvaldo Uchitel, and he said, very nice talk. And why there is many seconds delay between the high frequency stimuli and the full calcium response?
Probably because you have to generate a signal. We're looking at the nuclear calcium signal, so you have to generate a calcium increase at the plasma membrane that is perceived by the cytoplasmic ryanodine receptor that then propagate the signal to the nucleus. But if we look at the cytoplasmic calcium signal, the response is immediate. It's very fast.
So I have a question for you, Cecilia, and it's related with your glutamate and cage experiments. So these experiments were done engaging the glutamate on dendrites. So what are the mechanisms that are involved in that? Do you think that this glutamate is being released and acts autocrinally on the glutamate receptors that are present on the very same dendrites?
Well, I didn't want to go, it's already very long talk, so I didn't show that we check with a glutamate sensor, we checked that when we engage glutamate, the response was local. It only occurred at the site of engaging. It didn't go to the entire neuron. So we think that we are engaging glutamate that is stimulated with NAD receptors that promote calcium entry. And you can see, and I also did not show that there's an increase in the local calcium that propagates to the soma and the nucleus.
We have another question from Claudio Villalobos, and it is, what will be the biophysical mechanism in hippocampal neurons by which ryanodine receptor blockage decrease performance in spatial memory tasks?
Because we think that the generation of nuclear calcium signals is so important for memory processes, because it mediates the expression of genes that are important for the structural changes, for dendritic spine remodeling that occurs in memory processes. So we think that that's why it's so important.
And related to calcium signal, because you nicely showed that when you elicit calcium exit from the ER, so this calcium signal goes back toward the nuclei, where it activates the signaling cascade that you were mentioning during your talk. But it also, the calcium goes in the opposite way. It goes anterogradely. So what do you think is the role for the anterogradely directed calcium signaling?
We have not looked into that. It's a very interesting question, but we have not looked into that. Mm-hmm.
So we have another question here by the audience, by Pablo Lunardi again. And he said, I was wondering about controlling calcium level can be in some way, whether this could be related with calcineurin regulation, and this calcineurin could be associated to dendritic spine shrinkage.
Yes, very important question, and those are the things that we have next to study. We have to see, for instance, what happens to CAM kinase 2 and calcineurin when we have inhibited the ryanodine receptor function.
And since we have still a little bit of time, I have another question that is related with the redox part of your talk. And so you nicely showed that the ryanodine receptor have a redox component. So there's some system that can be oxidized. So what do you think is the mechanism that it's involved in the reversible reduction of those systems and how it is controlled?
Well, neuronal cells have enzymes that remove this. It's important to highlight that these are reversible redox modifications. So there's an enzymatic system that remove these modifications. And another thing that is important in terms of what you're asking me, Christian, is that when you age, and especially if you do not age very well, you have an increase in oxidation in neuronal cells. And it has been shown that that has some important effects, and we have done something in that regard too, in terms of frequency decodification. Because when you have an increase in the redox tone, in response to calcium entry, you have an increase in cytoplasmic calcium that activates potassium-activated potassium channels that after a sequence of stimulation, you have a long, prolonged after hyperpolarization phase that prevents neurons from decoding an incoming set of stimulatory stimulation. So it has been shown that disappears if you add antioxidant agents to the neurons. And also, we have shown that in aged rats, in the hippocampus in aged rats, ryanodine receptors are more oxidized and they are present at higher levels compared to younger rats. So this is all a recipe for disaster, because if you have increased oxidation and increased channels that respond to this oxidation, it will be very hard for neurons to regulate the calcium signals in response to stimulation. And it also in some neuropathologies like in Alzheimer's disease and in Parkinson's disease, there's an increase in the oxidative tone in the neuronal cells. So that also would contribute to having abnormal calcium signaling.
So we have space for the very last question, and this comes from Rafael Posada in Colombia. And the question is it possible that the ryanodine receptor induce bidirectional flow between mitochondria and ER? Bidirectional flow of calcium, I guess?
Yes, we have shown that in fact that occurs, that ryanodine receptor generates calcium signals that reach the mitochondria and increase calcium in the mitochondria. And again, I did not show that, but we have really demonstrated that not only IP3 receptors participate in the communication between ER calcium release signal and the mitochondria, but ryanodine receptors do so in neuronal cells as well.
Thanks so much. So we are coming to an end, and thank you very much, Cecilia, for a wonderful talk, very inspiring and with remarkable science. And so thank to all the audience to connect today and for the all insightful question that we received.
Thank you very much.